A big hello to all my wonderful champions. I hope every one of you who is watching this video is not at all stressed, but is just happy to find a smart way of studying. So yes, you know about hard work and hard way of studying. Now this is a smart way. Why do I say so? Because my dear children, whenever you are going to watch this video, there's 2 marker, 3 marker and 5 marker. Over here, we have found out the repeated questions. We are giving you the content. Now this content which is over here, okay, it might be reframed and given back to you on your day of your exam as well. So with 2 marker, 3 marker and 5 marker, that is the best part. If you have studied or went through all the previous year question papers, or if you know the model question paper questions, or if you have solved them thoroughly, you would be able to answer certain questions on the day of your exams as well. So which is why even if these videos, even if they go long, you have to say to yourself that I have to watch this because this could assure me what some good amount of marks. Do you get it? So yes, going ahead with the schedule that we have over here, this is for second PUC boards, my biology, my dear children, that is my subject. So Wednesday and Thursday is done. On Saturday, you would be getting model paper 2 and also this video, that is 2 plus 3 marker and also all diagrams. So reserve your Saturday for biology, my dear children. Lots of videos are going to come up there and you have to watch it. Do not miss even a single video out there because all of these are important things that you guys have only asked in the comment section. So even if today, if you want to ask something, whether it be exam related doubts or some subject related doubts that you have, you can ask. Okay, in the comment section, we are here to solve that up. And now on Monday, February 21st, you will be having biology all 5 marker, and Wednesday, February 26th, you would be having the passing package. Cool, let's go ahead. Okay, going ahead with order wise, okay, chapter wise, that is according to your NCERT. So your first chapter is sexual reproduction and flowering plants, and the weightage for this chapter is 11 marks. Now out of this 11 marks, you would be getting one 3 marker question for sure. Okay, I'm talking about the blueprints that was given, and according to the blueprint, there will be one 3 marker question for sure. Now 2 marker, it may or may not be there. So there are both the chances. We have to be prepared, right? So we have to solve all the 2 and 3 marker questions up there. So let's see. So complete the tabular column given below with respect to the male gametophyte of angiosperms. So what do you mean by male gametophyte? Male gametophyte is nothing but the pollen grain, because they are the ones that release the gametes. So you have to talk about them. What are the cells of the male gametophyte? So my dear children, first of all, you have to think about the pollen grain. In the pollen grain, there are 2 cells. One is vegetative cell. Okay, and this cell is very big, and in this big cell, no, there is another cell which floats, and that cell's name is generative cell. So now this answers the first part of the question, that is cells of the male gametophyte. One is vegetative and the other one is generative. Now the shape of nucleus of the cells. So vegetative cell which is there, it has an irregularly shaped nucleus. Okay, talking about the generative cell which is there, it has a spindle shaped nucleus. So you got your answer? Now I'll tell you one more extra thing. This generative cell is the one that will be dividing further and giving rise to 2 male gametes, which is why we call pollen grain as a male gametophyte, because it is the one that is going to give the gametes. So here are the gametes right over here. Now how do you frame your answer? You frame it like this, where you put vegetative cell and then you write generative cell. So I avoid writing on the board as much as I can, but yes. So shape of the nucleus, my dear children, it is irregularly shaped, and over here it is spindle shaped. Will you forget this? No, right? And here's the picture of it so that you remember it better. Okay, chalo. Going ahead. Differentiate between geitonogamy and xenogamy. What do you mean by geitonogamy and xenogamy, my dear children? So basically, over here, these are different forms of pollination. Okay, so geitonogamy is basically when you have 2 plants. Is it 2 plants? No. In geitonogamy, you just require 1 plant. Okay, so suppose this plant has 2 flowers. Okay. Here there is the female flower and here there is the male flower. From the male part, the pollen grains are going to come up over here in the female. Now this type of pollination which is occurring between 2 flowers but of the same plant, it is known as it is known as geitonogamy. Now on the other hand, if we have one more plant and then on this plant also we have certain flowers. Here it has a female part, and from here to here if it is going, my dear children, so it is between two flowers and it would be between different plants, then it is known as what? Xenogamy. So cross, you can remember like that. Xeno has X and X represents cross. Now in geitonogamy the thing can be, it already has this also, and this guy already has the female part. Still, if the pollen grains are moving from here to here, it will be still known as geitonogamy. Now what is the difference between geitonogamy and xenogamy? I hope you understood. This is between same plant, this is between different plants. Now genetic variation, which one of them will be bringing about genetic variation, my dear children? It's in the same plant, the flowers of the same plant. What genetic variation will it bring? It's more like asexual mode of reproduction, right? But still pollination is fertilization, everything is happening, so I cannot call asexual, but it is like that because there is no genetic variations between the same flowers of the same plant. If you talk about different plants, obviously this will have different genetic makeup and this will also have different genetic makeup. So obviously if it is happening like this, then there will be genetic variation. Okay, so how do I frame my answer? I can frame like this. Geitonogamy is a transfer of pollen grains from anther of one flower to the stigma of another flower of the same plant. Now genetically similar to the parent plant, this is the second point. So whenever differential between question comes, you have to write in tabular form. Please make sure you're writing like that. Okay, now here xenogamy, transfer of pollen grains from anther to stigma of what? Different plant, my dear children, and is genetically different from parent plant, brings variations. Now this is similar to autogamy if you talk about genetic sense, as in because it's happening between the same flowers of the same plant, which is why it is kind of like autogamy. Why? Genetically, only genetically I'm talking. In autogamy, in the same flower from the pollen grains to here it will be transferring. So genetically it will be same product that will be produced, it will be same like geitonogamy itself. Okay, both of them, they are cross pollination, but xenogamy is in an actual sense a true cross pollination that is being shown. So write these points and you get 2 marks. Now what is parthenocarpy? Give an example. Parthenocarpy is a very fun concept. So I'll tell you, usually what happens in the female reproductive part, no, there is ovary, and inside the ovary you have the ovule. So basically when fertilization happens, that is when the egg and the male gamete fuses, no, in here there will be zygote that is being formed in the zygote. So when fertilization happens, I would be calling this particular ovule as what? Seed. So I'll say this is getting transformed into seed. But what about the fruit out there? Sorry. What about the ovary out there? The ovary which is there, my dear children, it will be converting into a fruit. Okay, this is the usual case. So fertilization has to happen for this to happen. But in parthenocarpy, there is no need of fertilization. Without fertilization, still the ovary will be transforming into a fruit. So if there is no fertilization, do you see a seed over there? No. So these type of plants would be producing seedless fruits such as banana, my dear children. Now parthenocarpy can be induced. You can also make plants do this artificially. How? By injecting certain hormones. In that way also they will be ripening up. The ovary would be making the fruit, but there won't be any seeds up over here. You get it? So this is parthenocarpy. And how do you frame the answer? Ovary develops into fruit without fertilization. Such fruit is known as parthenocarpic fruit. This phenomena is known as parthenocarpy. Second point that you can write. There's a 2-marker question, so you have to at least write 2 points, right? Second point you can write is that parthenocarpic fruits are usually seedless. Are what? My dear children, seedless. Example is banana. And you can also write, you can induce parthenocarpy by inserting certain hormones. Now you might think, why does it naturally happen? Because of certain hormonal fluctuation that is happening in the case of plants. It can happen in humans, so it can happen in plants also, right? So that is the thing. Cool? Going ahead with the next question. I think I should pace it up a bit, otherwise it will take a lot of time to solve all the 2-marker and 3-marker. So if you agree, let's do it a little fast. Okay? So define the following terms, that is staminate flowers and pistillate flowers. What do you mean by staminate? Staminate comes from the word stamen, and stamen as in what? You're talking about the male part. You're talking about the male reproductive part of the plant. So those plant, or basically those flowers, okay, which just has the stamen, my dear children, which includes the filament and the anther, these type of flowers are known as staminate flowers. And the one that will be just having the pistil, what is pistil? Pistil or carpel is the female part, female reproductive part, my dear children. So in that case, they would be known as pistillate flowers. Cool. So how do you frame the answer again? Staminate flower, they are unisexual. There are no other sexes. There is only one sex, and that is male. Okay, they are unisexual flowers that bearing only the stamens. Pistillate flowers, unisexual flower, unisexual female flowers bearing only the pistil. Done? Easy? Okay. Next, sporopollen is one of the most resistant organic materials known. Justify. So where do you find sporopollen? So you studied about pollen grain, about the cells in the pollen grain, right? So the outermost layer that is there over here, it is known as plasma membrane. As any cell, it also has plasma membrane. Now outside the plasma membrane, there is a continuous layer which is known as intine, my dear children. I'm talking about pollen grain, and this is what? Intine. Outside the intine, there is something that is not continuous but discontinuous. What do you call this layer as? This is known as exine, my dear children. And exine is made up of very important substance which is known as sporopollenin, and this sporopollenin would help the pollen grains thrive through any condition, whether it be acidic, alkaline, there is high temperature, low temperature, salinity, low salinity, a lot of things. You get it? So the sporopollen which is there, it is the most resistant material which can let it survive during any condition. Now wherever sporopollen is not there, it will be making a pore, and this pore is known as germ pore, my dear little children. Okay, so how do you frame this answer? Sporopollen is one of the most resistant organic materials because it can withstand strong acid and also alkali. No enzyme that is there that degrades sporopollen is known so far. Every enzymes work on everything, break it down, but there is no enzyme over here. Now because of this property, pollen grains can be preserved as fossils as well. So because it stays for so long, you get it? That's crazy. Now sporopollen is found in the exine. I already told you that you have to frame your points. If you understand the concepts, you would be able to frame your points also really well. Okay, going ahead. Differentiate between pericarp and perisperm. What do you mean by pericarp and perisperm? So pericarp is basically, I spoke about the fruit, that is the ovary, right? So the outermost covering of the fruit will be known as pericarp. The outermost covering, or basically the covering of the fruit, is known as pericarp. Suppose you have mango. So mango, the outermost layer which is there, it is known as epicarp. Okay, which is like orangish yellow in color, and then the thing that we eat, okay, it will be known as mesocarp, and then the thing that we cannot eat, that is this, it is known as endocarp. Now these are the wall layers of the fruit. Together all of them, I would call it what? Pericarp. Now inside this there will be seed. The seed will also have its coat, that will be a different coat. Okay, that is known as seed coat. I'm talking about fruit, that is ovary. Ovary develops into a fruit, right? So these are the three layers that are being formed, and it forms a part of pericarp. Now you are asked, what is perisperm? Ma'am, I don't understand what is perisperm. So listen to me carefully. So when you talk about the ovary, right? The ovary has ovule, I told you. Ovule, in the ovule what happens is that you have so many cells. Making up something which is known as the nucleus. Okay, so what happens is that one cell among the nucleus will be the one that will be dividing further inside the ovule and giving rise to something that is known as embryo sac, where what will be there? Egg cell and everything will be there. So one cell divides and do all this drama. There will be still something left back of the nucleus. Okay, there will be still something left back of the nucleus cells. Usually it is degenerated or it has been used up, but sometimes it is left back. Why? To provide nourishment. Now this thing that is left back is known as perisperm. Okay, perisperm. Fine, will you be able to remember it? So you find perisperm in beet as well as bell pepper. Okay, so that are the two things that you need to know. So over here, black pepper, sorry, not bell pepper, it's black pepper and beet. In both of them, you would be having perisperm still persisting. So over here, how do you write it? So pericarp is the wall of the ovaries which develops into wall of the fruit and is known as pericarp. Epicarp is the outer layer, the skin of the fruit. Mesocarp, middle, that is fleshy in many fruits. Sometimes it's not fleshy, sometimes it could be fleshy as well. Endocarp, inner layer, hard or membranous, surrounding the seeds, I told you. Now, what is perisperm? The persistent nucellus is called perisperm, which is there for nourishment of the fertilized seed. Okay, sorry, not the fertilized seed, basically the embryo. It is for the nourishment of the zygote and the embryo. And it is found in black pepper as well as beet. Remember, these are very important examples. Going ahead, now we have a 3-marker question. The first 3-marker question, a very famous question that usually comes every single time. That is, draw a LS of grass embryo. What do you mean by grass embryo? Grass is an example of monocot, my dear children. They just have one cotyledon. Okay, so you have to label all this thing. So I will make you understand this thing and then we will be labeling a lot of things. Okay, so over here, listen to me. All the embryo, they will be having an axis that will be giving rise to the shoot, future shoot. So you call this as plumule. Okay, there will be something that will be giving rise to future root. You will be calling this as radicle. Now the radicle is covered by something that is known as root cap. You call this as root cap. Okay, now I told you they are monocot, that means they just have one cotyledon. So there will be one cotyledon right over here, dear children, and what do you call them? You call it as scutellum. This one cotyledon which is there, you call it as scutellum. Now, monocots are monocots, they just have one cotyledon. Why? Because the endosperm which is there, usually what happens is that we have endosperm. This endosperm is the one that makes two cotyledons in the case of dicots. Okay, if the embryo wants two cotyledons. But over here the embryo says, I just need one cotyledon, endosperm can still be there, no problem. So over here, little amount of endosperm still persist back. Okay, there will be little amount of endosperm that will be still there, and apart from that, on the plumule you will be having certain layers of things which are known as coleoptile, and over here also you would be having certain layers which is known as coleorhiza. Cool. So scutellum, coleoptile, shoot apex, okay, radicle, root cap, and shoot apex is basically talking about plumule. That's the only thing. So when you have to draw an LS, you have to draw like this, my dear children. Why? Because this is given in your NCERT. Ayo, yeah. This is given in your NCERT. So here there is a cotyledon, that is scutellum, my dear children. Here you have the plumule or shoot apex which is covered by what? The coleoptile. And then if you go down, there is radicle covered by root cap and also coleorhiza. And there is something that is epiblast as well. So draw like this, not like this, is just for understanding. It's usually how it works, but okay, they have made a little complicated, but okay, you can practice it and you would be acing it as well. Okay, so this is a 3-marker question, a very important diagram. You have to practice it anyhow. Okay, now next question, draw a labeled diagram of transverse section of young anther. Now this is very easy. How do you remember this thing? I'll tell you. So over here, young anther, you know, anther is bilobed, it has 2 lobes. So this is one lobe and there will be another one like this. Okay, that will be unequal, but it's okay. Now over here they have theca. So my dear children, understand that each of them will be having sporogenous tissue. Now this is where these cells are the one that would be dividing and further giving rise to microspores, and the microspores gives rise to, what do you call it as, pollen grains later on. So this is known as what? Sporogenous tissue, and this tissue is covered by something that provides nourishment to it. And what is that? What is that thing? That is known as tapetum, my dear children. Tapetum is the one that will be providing nourishment to the sporogenous tissue. Now, the outer layers, if you see, it has an outer layer which is known as epidermis. Now, inside the epidermis, it has another layer that is known as endothecium, and then in between this and the tapetum you have the next layer which is known as the middle layer. So you have to make 1, 2, 3, 4 layers, and then one big circle where the sporogenous tissue is also there. And that would be the transverse section of what? Your anther. This is exactly what they did over here. Okay, done. Chalo. Next, write any 4 characteristics of flowers pollinated by winds. What are false fruits? Give example. So I'd be telling it. First, I will talk about false fruits. Okay, false fruits, as in usually the ovary is the one that makes the fruit, but sometimes, suppose, okay, just suppose over here, you know, resting place is there, that is thalamus. On this thalamus, the flowers and petals and everything is there. Suppose instead of the ovary, this particular thalamus is also contributing to the formation of fruits. Okay, the thalamus is also contributing to the formation of fruits. You would be calling this fruit as false fruits, because usually ovary is the one that takes the whole credit for making the fruit. Over here, thalamus is also contributing, or it's not just thalamus, any other parts apart from the ovary, if it is contributing for the formation of fruits, you will call it false fruit. Example, the apple that you eat. The apple that you eat, you basically get it from both the ovary as well as the thalamus. Okay, now we'll talk about the 4 characteristics of flowers pollinated by wind. So first thing, wind pollination requires that the pollen grains are light in weight. Otherwise, if they are moto, will they be able to move around very easily? No. So they have to be light in weight so that it can be easily get transported. Now, they should be non-sticky, my dear children. They should be what? Non-sticky, because if they go and stick to something, will they be able to move? No. This is all logical things, okay. Now, they would be often possessing well-exposed stamens, okay. There will be well-exposed stamens such that when the wind comes, they can basically send their pollen grains as well, which is why they have a well-exposed stamens. Now, to receive the pollen grains, there should be what? Feathery stigma. There will be feathery stigma which captures all the airborne pollen grains which are there. Now, the wind pollinated flowers often have a single ovule. Why? Because wind pollination is kind of, you know, it does not guarantee pollination. So, if you make multiple ovules and keep it, you basically wasting a lot of energy, right? So often they have only a single ovule in each ovary, and they would be having numerous flowers packed into what? Inflorescence. There would be so many flowers which would be packed into inflorescence. Okay, cool. Works so that any one of them at least can trap. So these are the things that you need to write for the first question. False fruits developed from the parts other than the ovary, like the thalamus or the pedicel. Example, you have apple, that is from the thalamus, cashew occurring from the pedicel, and strawberry as well. These are the examples. And now we would be reading the answers as such whenever the answers are given, so that it saves a lot of our time, because it's already 22 minutes for one chapter. Yes. So describe the structure of dicot embryo with the help of label diagram. So whenever I think of doing something like that, a diagram question comes. So listen to me. I told you dicots, as in they have 2 cotyledons, my dear children. So they will also have an embryonal axis which looks like this. So they will be having a plumule. Okay, they will be having a radicle, and they will be having a root cap. Okay, now this part, which is now over here, my dear children, they will be having 2 cotyledons. This is suppose I am making like this. This is one cotyledon, and this is the second cotyledon that is there. Now, the part, the embryonal axis above the joining of the cotyledons, you call this region as epicotyl, and below it you call this as hypocotyl. And this is basically the cotyledons. Now, when you make it, you have to make it like this. So, I am going to show you, listen to me carefully. Okay. Just a second. Okay, so basically, this will be the plumule, and over here these are the cotyledons, and then you would be having the radicle with the root cap. So this is the picture. How does it look? Okay, so this is a plumule, the cotyledons, the hypocotyl, the radicle, and the root cap. So not to confuse you, which is why I probably made it something like this. Even though it's funny, it will be helpful for you to understand the diagram at least. Yeah. So it consists of, you have to describe the structure of with the help of label diagram, but you can support your answer with 1 or 2 points. Okay, 1 point would be more than enough. So it consists of an embryonal axis and 2 cotyledons. Embryonal axis as in from where you get the shoot and the root. Okay, now the portion of embryonal axis above the level of cotyledons is epicotyl, which terminates with plumule or stem tip. Okay, now the cylindrical portion below the level of cotyledons is hypocotyls, that terminates at its lower end in radicle or root tip. The root tip is covered by root cap. And with that, we covered that particular question as well. And you know what? We have covered sexual reproduction in flowering plants. A big kudos to all of you who have watched until now, and a big kudos, give a tap. Okay, because you guys nailed it. Okay, 22 minutes of one chapter, and we are getting 2 or 3 markers from this. Now over here, if you are someone who needs to know about the schedule, so here is a schedule. If you're not in the WhatsApp group, basically for PDF, everyone asks, where is the PDF, where is the PDF? Array, you get the PDF in the WhatsApp group. Join the WhatsApp group. Okay, you can join the WhatsApp group, you will get the PDF as well. Now over here, this is the preparation schedule for mathematics. If you want to take a screenshot, you can take a screenshot, right? And this is for physics that you see over here, and this would be for chemistry that you are seeing over here. So take the screenshot and do the best preparation. You are gonna ace, my dear children. Now going ahead to the next chapter, shall we go ahead? So it is human reproduction, which carries either 11 marks or 10 marks. So according to one blueprint, it carries 10 marks. According to 2 other blueprint, it carries 11 marks. Okay. Now there will be one 3-marker question for sure coming up from this particular chapter. That is for sure. So according to 3 blueprints that are given, there is one 3-marker. So let's start with the first 3-marker. Parturition is induced by complex neuroendocrine mechanism. Comment on it. Any idea? What is it? So basically, I'll tell you a story. Okay, suppose I am the baby. I am what? The baby. Now, I am in the uterus. So from my body, certain hormones will be released at the time of when my whole, growth and everything has been done. What is this hormone? These are androgens. Okay, these are androgens. Now, if you do not remember, it's okay. At this point of time, what happens? Basically, I am inside the uterus. Yes. I am inside the uterus, and what happens is that in the uterus you have placenta. So placenta will be releasing lots and lots of estrogen, my dear children. It will be producing lots and lots of estrogen. And what does this estrogen do? You know, we have a layer that is known as myometrium. Okay, we have myometrium, perimetrium, outer layer, myometrium, middle layer, and then you have endometrium. In the myometrium, there will be so many oxytocin receptors which gets active. Because of this rapid estrogen production. By what? By the placenta. By the placenta. Okay, and the baby is the one actually producing androgens. Baby is the one that actually produces androgens. What does the placenta do? It converts the androgens to estrogen. And this estrogen says that, hey, let's make lots of receptors ready over here. Now, the baby, that is me, okay, when I would be moving down, okay, I would be moving down the uterus. What happens? When I reach over here, the baby is basically pushing, right? So there will be so many nerve endings over here. Which gets activated. Now, which gets activated? Until now, my dear children, I spoke about hormones. Now I'm talking about nerves. So listen to me carefully. When the baby tries to move out, what happens? There will be so many nerve endings that gets activated. Now, this will give a nervous signal to the maternal pituitary gland. This will give a signal to the maternal pituitary gland. Until now, did you understand? So this placenta releasing estrogen and making so many oxytocin receptors active is hormonal. Over here, the baby moving out, okay, baby trying to move out and giving a nerve impulse over here, my dear children. This is nervous response. And together, you call this both thing, okay, the placenta and the baby thing together, you call this as fetal ejection reflex. So it's the baby and the placenta that gives signal that we have to move out anyhow. Now this signal goes to the maternal pituitary. From the maternal pituitary, who is going to be released? Oxytocin is going to be released. Now the oxytocin will come over here and see that, hey, I have so many receptors. Why can't I just bind over it? And it binds over it. And what happens, my dear children? Whenever it binds overhead, the smooth muscles in the myometrium, they will start contracting. When it gets contracted, I won't have space, so basically I would be moving down. You get it? So that is the whole thing. And this is why parturition, we say it is induced by complex neuroendocrine mechanism. Do let me know if this type of explanation is helpful for you. Otherwise, I would stop giving this type of explanation. Okay, you feel that's lots of waste of time and all. You will do let me know. But in this way, I think children remember for a better or longer run. Yeah. So anyways, So triggered by cervical stretching. So when the fetus reaches the full term, its head pushes against the cervix, activating certain receptors, stretch receptors. Now the nerve signals is sent to the brain. The stretch cervix sends signals to the hypothalamus. Now the hypothalamus will tell the posterior pituitary to secrete oxytocin in response. This oxytocin will stimulate powerful contraction in the uterus. More contraction lead to more oxytocin release, ensuring labor progresses until birth, my dear children. So before that, you have to talk about a bit about what estrogen also. So say that estrogen is being released by what? The placenta, okay, which makes so many oxytocin receptors active on myometrium, and then you can talk about all this thing which is there. Okay, so this is the fetal ejection reflex that is coming, my dear children. This plus this plus this. The first point is fetal ejection reflex, and then the whole thing happens. So you have to tell why it is not neuroendocrine, because over here both the nerve impulse, neuroendocrine mechanism as in when both the nerve impulse as well as the endocrine, the hormonal system is being involved in the process. Then you start off your answer with this, write in points. Okay, chalo. Next, in the figure given below, part A and part B show levels of hormones which influence the menstrual cycle. Study the figure and answer the questions that follows. Okay, so are we ready? Chalo. Name the organs and glands which secrete the hormones represented in labeled part A and B. So over here, if you have seen the menstrual cycle, my dear children, you might have seen these hormones FSH and LH, estrogen and progesterone. Okay, so this might be very dark because of the light ratio. Anyways, let me tell you. So estrogen and progesterone which is there, it is released by the ovary. Are you listening? It is released by the ovary. FSH and LH are pituitary hormones. They are produced by anterior pituitary. You know, oxytocin is released by posterior pituitary. Pituitary had two parts, two lobes, posterior and anterior. Okay, so anterior one produces FSH and LH, and also ovary produces what? Estrogen and progesterone. So name the organs and glands which secrete the hormones represented in label part A and B. What will it be? So it will be, A will be what? Pituitary gland. B is what? This gland, that is ovary. Okay, cool. Now state the role of hormones secreted from part B on the uterus of human female during the menstrual cycle. So what is the role of estrogen and progesterone? That is what is being asked. So do you know the role? I'll tell you. First thing, listen. First is the menstrual phase. In menstrual phase, I will be going really fast, okay? Listen to me carefully. In the menstrual phase, what happens? The endometrium which is there, it is now going to shut down. It is now going to shut down. Why? Because fertilization didn't happen. But our body does not give up. It starts reforming the endometrium. Why? Because estrogen is being produced. Estrogen is being produced. Where, ma'am? How? Basically what happens? The endometrium is shutting down in the first phase, that is menstrual phase. At this point of time, the follicles, the next follicles, okay, they would be getting matured. Now these matured follicles, they will be releasing certain estrogen, and the amount of estrogen, when it goes high, what will happen? The endometrium will just shut down. Now it will be, new layer of endometrium will be slowly forming up. Okay, it will be slowly forming up in this phase, that is proliferative phase. This is known as proliferative phase. It is also known as follicular phase because the follicles are also developing. Now one level it will reach, that is on the 14th day, what will happen? The follicle will be rupturing. Okay, the follicle ruptures, the ovum will be released. Sorry, not the ovum, the secondary oocyte which is there, it will be released. Now, Graafian follicle is the one that ruptures. Now, whatever thing is left back in this particular follicle, it is known as corpus luteum. And the corpus luteum will be releasing two hormones, that is estrogen and progesterone. Again progesterone and estrogen. Okay, so what will happen? Until now progesterone was not there, but now since because of the corpus luteum, the amount of progesterone will be way too much, and now estrogen also goes way too high. So at this point of time, the progesterone and the estrogen will be thickening, especially the progesterone, my dear children, which you see over here. It will thicken the endometrium lining really well, and it will make sure that shedding of the endometrium is not happening. So initial is done by estrogen, and then the later one is done by progesterone. Okay, now if fertilization is not occurring, both the hormones, my dear children, their level will be dropping and the menstruation will be happening, that is blood flow will be happening. So estrogen in the proliferative phase stimulates endometrial growth and thickening, prepares the uterus for implantation, such that if the egg and the sperm meets, both can come over here, that is the zygote can come over here, form an embryo and then later implant. Now progesterone, what does it do? Mainly it is secreted during the secretory phase. It does not mean it is not it's secreted in the next phase, okay? Estrogen is also secreted. So now over here, it stabilizes, maintains the thickened endometrium which is there and enhances the glandular secretion, okay? That is, so many secretions will be coming out of the endometrium such as glycogen and things like that. Suppose if an embryo comes, it has to provide nourishment, right? For that it is being done. If fertilization does not occur, both the hormones level go down, leading to menstruation. So this is how you need to frame your answer for a 3-mark question. Now, during pregnancy, the level of hormones are increased several folds in the maternal blood. Name the hormones and write their importance. So first thing, during pregnancy, you in your body, one very important thing is being formed, and that is known as placenta. So obviously there will be 4 placental hormones only that you cannot forget ever. That is what? HCG, my dear children, human chorionic gonadotropin, and then you have HPL, that is human placental lactogen, and then estrogen and also progesterone. So these 4 hormones are for sure being released during what do you call it? What was I saying? During pregnancy for sure by the placenta. Apart from that, you know, basically you have cortisol being increased, you would be having prolactin which be increased, you would be having thyroxine whose level will be increased. So all these things are increased, my dear children. And also by the ovary, a hormone is being produced which is known as relaxin, but this is more towards the end of the pregnancy. Towards the later phase of the pregnancy, the ovary releases this. So it's the ovary that usually releases estrogen and progesterone, but placenta also does. You have to remember it. Okay, now at least these things should be there, and the names of all cortisol, prolactin, thyroxine should be there. Thyroxine is for metabolism, cortisol is for forming sex corticoids and things like that. Prolactin basically would be helping the mother to build up the mammary gland really well, okay, because she is going to lactate, right? She is going to breastfeed. So for that. Now, what is the role of HCG, human chorionic gonadotropin? So I was talking about this corpus luteum. Okay, that is the things that you get from the Graafian follicle, right? Graafian follicle rupture, you got the corpus luteum which was doing all the stuff. Now the corpus luteum, to maintain it, there should be someone to maintain it. Otherwise what will happen? The corpus luteum will degenerate. So to maintain the corpus luteum so that it's continuously produces the progesterone and everything. You need progesterone for endometrial lining maintenance, right? So for doing that, you have HCG. HCG tells that, hey, corpus luteum, you have to be there. You have to still produce progesterone and everything. So it is secreted by the placenta, maintains the corpus luteum, ensuring the production of progesterone during the early stages of pregnancy. Now progesterone produced by the corpus luteum and later the placenta, it maintains the uterine lining. Estrogen secreted by the placenta promotes the uterine growth. Human placental lactogen produced by the placenta regulates the maternal metabolism and also helps in what production of in the growth of the mammary gland and things like that. Relaxin, it is produced by ovary during the later phase of pregnancy, softens the cervix and relaxes the pelvic ligaments. Why to relax? Basically to provide space for the baby to move out. Okay, so it is done during the later phase of pregnancy. And also, my dear children, something that is much more important, cortisol, okay, the name, and then thyroxine, okay, and as I said, one more thing, prolactin. Someone is calling me. Okay, so these 3 things are really, really important. You have to write it. Now, list the functions of human placenta. I just told you. Okay. The functions of human placenta. So, placenta is found between the mother and the embryo. It is formed with the help of both the embryonic tissue as well as maternal tissue. Both of them, they contribute to form the placenta. Now, why is placenta important? Placenta will be helping in exchange of very important gases such as oxygen to the baby and carbon dioxide outside the baby. Okay, so basically for removing the carbon dioxide, for removing the waste materials also, okay, and for providing nourishment to the baby. So these are the functions of human placenta. You can write like this. Placenta connected to the embryo through an umbilical cord which helps in the transport of substances to and from the embryo. So it's not like, suppose this is the baby with the placenta over here, my dear children. So the placenta is connected like this with the help of umbilical cord, and it's not like a free space where things are getting moving. No, these are having blood vessels, and the blood vessels are going to come up over here. From here, diffusion of oxygen, carbon dioxide and everything is going to happen. Okay, so placenta facilitates the supply of oxygen and nutrients to the embryo and also removal of carbon dioxide and excretory waste. Okay, supply of oxygen and what? Nutrients, removal of carbon dioxide and excretory waste. Now it also acts like an endocrine tissue. This is a very important point because it releases all the hormones for the maintenance of pregnancy, right? Human placental lactogen, like human placental gonadotrophin, human chorionic gonadotrophin, okay, human placental lactogen, estrogen and progestogens. Cool. With that, we complete that question as well. Now, briefly explain sex determination in the case of human beings. In the case of human beings, how is the sex determination happening, my dear children? The males will be XX, the females will be XY. Agreed? Okay, so these are the parents. Now, over here, the gametes that will be formed. From a pair of chromosome, only one can go into one particular gamete. So, this is one and then this could be the next one. Cool. So either this X chromosome in this gamete, X chromosome in this gamete. Here the male will be producing two type of gametes, either X or Y. Now suppose the egg fused with this type of sperm, so you get XX. This is going to be a female. If this fused with this, you get XY. This is going to be a male. This with this XX, again a female. This with this XY, this is going to be a male. This is how sex determination happens in the case of human beings, and there are 50% of chances that the kid will be boy, the 50% of chances that the kid will be a girl. Cool. Unless and until there are some genetic problems. Now, distinguish between menstrual cycle and estrous cycle. You know, menstrual cycle happens in the case of primates like us. Our brain capacity, everything is very much, okay. There are non-primates as well. For example, you have what's a dog, a cat, cow, all of them. So they do estrous cycle. They have what? Estrous cycle. And in them things are different. For in the case of menstrual cycle, basically once it starts, it goes off until the age of 50 or something like that. You get it? It is continuous process. But in the case of estrous cycle, my dear children, it happens sometimes, okay, in one particular phase. It is kind of seasonal thing that happens. And over there, one more very important difference is that in our case, when the endometrium is there, it sheds off, right, in the form of menstruation. Over there, the endometrium is reabsorbed. The endometrium is reabsorbed if fertilization is not happening, which is why dogs and all, you would not find them bleeding. Spotting can happen, one or two few drops of blood can be there, but mostly they would not be bleeding like human beings. Why? Because they show estrous cycle. So what are the little differences? This is the little difference. So it occurs in primates like humans and monkeys. Over here, non-primates like cows, dogs, etc. Menstruation, endometrium lining shuts if no pregnancy. Here no menstruation because endometrium is what? Reabsorbed, my dear children. Sexual activity, females are sexually active throughout the cycle. Okay, now here sexual receptivity only during the estrous period. There is a phase, my dear children, there is a phase in their cycle. Only during this particular period their sexual receptivity will be there. Now frequency, typically monthly. Okay, every month this cycle will be occurring. Over here can be seasonal or can be continuous. Can be seasonal as I told you in the case of dogs and horses, or it can be continuous as in the case of cows as well. Now ovulation, when does the ovulation happen? It happens during mid-cycle. Suppose it is happening every month. So every month middle there will be ovulation happening in the case of human beings. In the case of estrous cycle, by dear children, whenever they have the cycle, it is seasonal or can be continuous. Either way. So whenever there is a phase that is estrous phase, that's when the ovulation would be happening. So these are the differences that you need to write for a 3-marker question. Okay. Now in NCERT, one small thing is given and they are putting out a question out of it. That's kind of bad, but yes. Shall we go ahead with the next question? Yes. So we have reproductive health, my dear children. It is a 6-marker chapter. So either 2-marker or 3-marker can come, or either 1 2-marker or 1 3-marker can come, or both of them can come. For one each can be happening. So we have to be sure of this. So mention the 2 medical grounds on which pregnancy are subjected to termination. Why do you say it is okay to terminate the baby? When do you say that? That is the question. And the answer goes like this. The 2 medical grounds on which pregnancy are subject to termination are: if continuing the pregnancy poses a risk to the physical or mental health of the mother, risk to the mother's health, or if there is risk of fetal abnormality, as in the baby is having some genetic disorder, and if at all it is born, it would not be able to have a good life, then that case also they are recommended to undergo medical termination of pregnancy according to the MTP Act. Okay, so if there is a substantial risk that the fetus may be born with physical or mental abnormalities, making it incompatible with life, at that point it is okay to do medical termination of pregnancy, otherwise it is not. Okay. So please write these 2 things. Now what, okay, so especially risk to mother's health, and also if it is a first sex, that is in the case of rape also, things can be, MTP can be done. Okay. Now, what are emergency contraceptives? Right there, hormonal combination. Emergency contraceptives, as in, again, is a forceful rape or forceful sex, as in rape, okay, or any such condition arises. At this point of time, obviously no one would want to undergo pregnancy, right? So at this point of time they are taken emergency contraceptive, which could be immediately preventing pregnancy to happen. So what are the things that you put over here? Basically you take a hormonal combination of progesterone and estrogen. So usually in our body, progesterone and estrogen basically helps in the maintenance of pregnancy. But when the level is increased, my dear children, they would be doing the opposite work. They would not allow the endometrium lining to be there. They will make it very thin so that the embryo does not go and implant. Okay, they would be bringing out lots of changes in the uterus such that it becomes totally incompatible for the embryo to get implanted over there. And sometimes it does not allow ovulation to happen. Okay, it does not allow ovulation to happen. Okay, that is one thing there. So basically forceful rape happened. The sperm entered the body. The sperm can survive for some days. So this term, some days, if ovulation happens, then what will happen? So to prevent ovulation also, this particular thing is being given. To prevent ovulation, to prevent fertilization, to prevent implantation, for all the things, the hormonal combination of estrogen and progesterone is given, usually within 72 hours. So pills are used to prevent pregnancy after unprotected sexual intercourse or contraceptive failure. Okay, if the contraceptive method that you are using, that is also failing, at that point this is used. Now they are intended for use within a short window, that is usually within 72 hours after the event. Administration of progestogens or progesterone-estrogen combination or IUDs are being best for emergency contraceptives. Cool? Chalo. Now what is infertility? Give reason for infertility in humans. Infertility as in without unprotected sex, still they are, the male and the female, they are not having a fetus or a baby. This is known as infertility. Now give reasons for infertility in humans. This can be many reasons. So infertility is defined as inability to produce children by couples in spite of unprotected sexual cohabitation. The reason for this could be physical, congenital, or any diseases, or the drugs that they are using, or immunological, or even psychological. Okay, there are so many reasons for infertility to happen. So there can be less sperm count, the ovum, that is also there. Okay, the ovum quality is not that great, the sperm quality is not that great, the sperm count is not that much. Okay, the semen will be there, but the sperm count won't be not there. So all these reasons could be something that is leading to infertility, or even the uterus, it is not allowing for implantation to happen. Okay, so these are the things. Now, what are the features of an ideal contraceptive? Mention the natural methods of contraception. So, ideal contraceptive, how should a contraceptive should be? First thing, it should be user friendly. Everyone should be able to use it by themselves. Second thing, it should be available easily. Okay, it should be easily available, it should be affordable. Okay, now also it should be reversible. The action should be reversible. Apart from that, what are there? Okay, effective and reversible, and also should not interfere with the sexual desire of the couples. So these are the things which comes under ideal contraceptive. And just give me a second, someone came in. Okay, so over here, natural methods of contraception, as in there is nothing that we are using from external sources. Okay, first thing is periodic abstinence. What do you mean by that? That is, every woman would be having a fertile window where there are higher chances that ovulation would be happening and that ovum will be released. At this point of time, obviously, if the couple would be having sexual intercourse, there are higher chances that fertilization could happen and the baby would be there. So basically, periodic abstinence is resisting themselves from having sexual intercourse between the fertile window. Okay, now then you have withdrawal method or coitus interruptus. That means, again, same thing. Basically, during the process of sexual intercourse, the male would not be ejaculating inside the female body, instead would be withdrawing the penis from the female body, and that is known as withdrawal method. In that way, the sperm would not be released. Now, lactational amenorrhea again is a natural method. Why? Because the lactating mother, the breastfeeding mother, my dear children, would not be having menstruation for 6 months after the baby's delivery, because she would be actively lactating. At this point of time, the prolactin hormones are way too much in the body, which is why ovulation was not going to happen, and also menstruation would not be happening. So these are the natural methods of contraception mentioned over here. Okay, going ahead with the next thing. The popular and effective contraceptives include IUD. Mention the type of IUD with an example of each. So IUD is intrauterine devices, as in devices which you are going to put inside the uterus, my dear children. So first one is in the IUDs that is there. Okay, I think I have put it. Yeah, first one is non-medicated IUDs. That means they are not going to release anything up there. Example is lipase loop. How does it work? Basically, it is going to act like a foreign particle in the body. And now when something foreign comes inside the body, what will the body do? It will react and will make the uterus unsuitable for implantation to happen, and also the sperm would not be able to enter or basically reach the egg because it is acting as a physical barrier in the uterus. Now the second thing is copper releasing IUDs, my dear children. Example is copper T and also copper 7. Okay, or multilode 375, that is also there. Now over here, these IUDs are going to release copper ions. And what does this copper ion do? It basically goes and tells the sperm that you cannot move around here. So it messes up the motility of the sperm. Now if the sperm are not motile, they would not be able to go up to the egg and fertilize it. Okay, so it will be really toxic for the sperms, and they also do phagocytosis of sperm. What do you mean by that? They kill the sperms. So that is also being done. This also prevents what's fertilization. Now hormone releasing IUD is there. That means these IUDs, when they are inserted, they are going to release hormones in the body. Example, progesterone and everything is being released. Now example of this is what? Progestasert or LNG20. LNG20 or progestasert, not get confused with, do not go right here LNG20. Okay, LNG20 will be here itself, and progestasert is also here itself. It is going to produce progesterone or levonorgestrel. Okay, anyways, they would be thickening the cervical mucus. What do you mean by cervical mucus? So over here in the uterus, you have the cervix region, right? The cervix region over here, if the mucus plug is being formed, do you think the sperm which is there, it can enter the uterus? No. So that is being done. Also, it inhibits the ovulation and makes the uterus really unsuitable for implantation. How am I not able to pronounce this? Levonorgestrel, something like that. I forgot the pronunciation all of a sudden. Okay, so basically these are the things that you should be knowing. Going ahead, and I'll tell you, IUDs are the most recommended thing for female if they want to delay the pregnancy or have spacing between their children, and it is the most accepted method of contraception. This is also given in NCERT, a very important point for your MCQ. Going ahead, so just 3 simple principles through which we can prevent sexually transmitted diseases. So obviously first one is practice safe sex, that is always use condoms or physical barriers. Basically condoms during the sexual intercourse to reduce the risk of STDs, because STDs are transferred with the help of fluids, body fluids such as genital fluid as well. So if condom is there are high chances the genital fluids would not be mixed. Now maintain monogamous relationship. Obviously, if there are multiple partners, there are higher chances that you can contradict STDs from any person that there. Now, avoiding sharing needles. Now, this is not exactly, a way to prevent sexually transmitted diseases, but I'll tell you why. So such diseases like HIV and hepatitis, they are spread through body fluids, which includes blood as well. So if you're someone who is using the same needle as the other person, what will happen? You might also contradict this particular disease, my dear children. So you may or may not put this point over there. If you're mentioning, you have to tell that sexually transmitted diseases like HIV and hepatitis B is also spread through blood, which is why avoiding sharing needles could also be an option. Apart from that, obviously you should be meeting a counselor, talking to them. In that way you can prevent a lot of things in your life. Okay, so those are the things given in your NCERT. This is not something that I said. Yes. Now, what is contraception? Briefly explain 2 barrier methods and 2 surgical methods. 2 barrier methods, as in it is going to act like a physical barrier between the sperm and the egg to meet. So one such method is basically a condom. Okay, one such method is, why is natural method giving over here? Just a second. So barrier method is written and natural method is given over here. So you don't have to write this. Okay, barrier method, as in you can talk about condom. Okay, there is male condom as well as female condom which can be used. Apart from that, there are diaphragms, my dear children. There are diaphragms. Okay, just a second. Oh my god. Okay, diaphragms, and then you have valves being used. So whenever you use waltz and all, waltz is basically inserted into the cervix, such that the sperm would not be moving up. So now whenever you use that, you make sure that you're using jellies or spermicidal things, so that the sperms which are there, they could be killed as well. Okay, so these are the methods of contraception. Obviously, you do not want the sperms to get inside the female body. We get it? So physical barrier method, you have this thing, that is condoms, as well as you have diaphragm, balls and everything that I explained it to you right now. And surgical methods, there are two, my dear children. One is tubectomy and one is waxectomy. In tubectomy, you are going to cut the fallopian tube. I'm going to tie it there, because fallopian tube is the one that helps in the transport of ovum, my dear children. So if you're cutting it, obviously the ovum would not be transported further. Does that mean the ovary would not be releasing ovum every month? The ovary will be definitely releasing ovum every month, my dear children. So that person will always having menstruation also. That person would be having menstruation, just that the ovum would not be able to reach. Okay, the ovum would not be able to reach the entire position of the fallopian tube, because it is being cut over there. So it cannot meet the sperm. That is the only thing that is happening up over here. Now that is a surgical method. This is irreversible. Okay, now talking about vasectomy, it's when the vast difference of the males. Now in males, you have so many accessory ducts that carries the sperm. Now vast difference is the the longest one out there, not the longest one exactly, but yes, it is the one that will be carrying things from the testis to the urinary bladder, right? So now over here, the vast difference is cut and tied. Same thing, it will prevent the transport of sperm. But does it prevent spermatogenesis happening up over here? Sperms would still be formed. You have to remember, sperms will still be formed, and this person will be releasing semen also, because it is the accessory glands. You know, accessory glands, they would be releasing certain secretion, thinking that sperm will come. So basically there will be seminal plasma that will be still released, and over here spermatogenesis is still happening, such that the only thing is that the semen which is there, the semen that is going to be released, would not be having sperms. Actually, it is very wrong to say that the semen is being released. Semen means sperm plus seminal plasma. Semen means sperm plus seminal plasma. But over here only seminal plasma is being released, there is no sperm. So you cannot call it semen, but inside it is written that semen is being released without sperms. So you have to follow that. Okay, so these are the two surgical methods, and with that we complete that chapter as well. And now we go ahead with the highest weighted chapter, that is Principles of Inheritance and Variation, carrying 13 marks, and here you can expect 1 2-marker question for sure. Okay, so are we ready? Yes, I hope so. Let's get into it. So write a short note on sex determination in birds, my dear children. So in birds, there's a very unique way. Okay, so usually we see XX. Okay, females, they'll produce same type of gametes. Either they are producing just XX in females. In our case also, all the gametes that they are going to produce will be having an X chromosome. But over here, there's a twist. That is, the females that is there, my dear children, they will be having ZW. Okay, they will be having a genotype of ZW, while the males will be having ZZ. Now, what do you call this? I'll tell you. Okay, so here the gametes, when they are going to form from both the pair, one can be going into a gamete. So these are the gametes of the males. So the male gametes are similar. So I call this condition as homogametic condition of males. So male homogamete is seen over here. What about females? Females produce a gamete like this or like this. So these are heterogametes. So there are two different type of gametes being produced. So this is heterogamety. Female heterogamety that is being shown up over here. Now suppose this one fuses with this particular egg. The product will be ZZ, that means it will be a male. This fuses with ZW, so it will be ZW, it will be a female. Now this with this ZZ, this with this ZW, again this will be what? A female, my dear children. So this is a method of sex determination in the case of birds, where the birds, they show female heterogamety, my dear children. So you have to follow through all the things that are written up over here. So just go through it once and it will be all clear. Exactly what I have said. Okay, so in this, female is heterogametic and male is homogametic. Female produces two type of eggs carrying Z and W chromosome. If the sperm carrying Z chromosome fertilizes the egg carrying Z chromosome, the zygote develops into male, whereas if it carries Z chromosome and the other one carries W chromosome, then it will be a female. All these things. Okay. Now, going ahead, so it is known as ZZ-ZW type of sex determination. Now, what is haplodiploid sex determination mechanism? Mention an animal which shows this. So, haplodiploid sex determination, as in, if it is diploid, as in if it has 2n number of chromosome, normal number of chromosome, this will be a female. If the zygote has half the number of chromosome, that is haploid, then it will be a male. Then it will be what, dear children? It will be a male. This is known as haplodiploid sex determination. Where do you see it? You see it in honeybee. You see this in honeybee, and honeybee is the only example given in your NCERT, so you have to remember it. So usually it is said that insects have a different way of sex determination, that is exotype of sex determination. Please remember honeybee over here is an exception for that. Now listen to me carefully. Okay, you have to listen to me carefully, and that is not actually an exception for that, but yes. So you particularly say honeybees show haplodiploid sex determination. Now I am going to tell you why. Listen. So it is not actually an exception, this is the actual case, exotype of sex determination only, but how? I'll explain. So over here, suppose there is 2n. There is what? 2n. So when a cell is there and it is going to make the gametes will be n, my dear children. It will undergo meiosis, right? It will undergo meiosis, and the gametes that will be produced will be having haploid number of chromosomes. Over here, it will be undergoing mitosis, and it will be undergoing mitosis, not meiosis. So it will also be haploid. Suppose this and this came together, you get something that is diploid, then it will be a female. Suppose this is not undergoing fertilization, then this will be what, my dear children? It will be a male. Okay, got it? Now, over here, let's see that. Haplodiploid sex determination. So female is 32 and the male is 16. 32 is the diploid number of thing. Okay. So when they are going to form the gametes, okay, one gamete will be having half the number of chromosome, another gamete will be having half the number of chromosome. Here also the male will be just transferring whole number of chromosome which is there, because it is mitosis, my dear children. Now over here, suppose this and this gamete fused, it will be 32, it will be female. If this does not undergo any fertilization, this is known as parthenogenesis, where the unfertilized egg is going to become what? A male, my dear children. So haplodiploid sex determination mechanism is a system in which sex of an individual is determined by the number of chromosome sets it inherits. So male develop from unfertilized egg and are haploid, having single set of chromosome. Females develop from fertilized egg and are diploid. A queen bee lays egg. If an egg remains unfertilized, it develops into male drawn. Okay, if it is going like this, it will become a male. That's my hair. Okay, it will become a male drawn. And if an egg is fertilized, it develops into female worker. This would be a female worker. There won't be any more queues that will be coming up along. Okay, there are very chance of a queen to be formed. Queen will be there for so many years. Okay, now depending on environmental and dietary factors, it will depend. Okay. So you give the worker bee something, okay, royal food, royal jelly you call it, okay, which is why it will turn into a female. Sorry, not in a female, basically a queen. So I'm not getting into that because it will be too confusing for you guys to understand, and I did something, so just wait for a second, okay, and I'll sort it out and come. Okay, going ahead with the next question. Write the genotypes of parents when their children are with A, B, and O blood groups. So many blood groups are there. So we have to find out. Children are having so many blood groups, my dear children. So when do you get A, B, and also O blood group? That means both the parents should have at least one A, and the other parent should have at least one B. O would be only shown if both the parents would be having one O. And AB will also be showing if both the parents have one A and the other will be having one B. So in this case, suppose the one parent is like this, another parent is like this. Now when the gametes are formed, it can be IA, or it can be IO, or it can be IB, or it can be IO. Now suppose this and this fuses, you get IA, IB. That means it is AB blood group because both are codominant, my dear children. Suppose this and this fuses, you get IA, IO. Now this is A blood group, right? Now because there is this one to dominate over O. Now suppose this and this fuses, you get IB and IO. In that case, B will be dominant, so this will be B blood group. And this fuses, that is IO. Over here, there is no one to dominate, so it will be O blood group. In that way, we get all the type of blood groups, my dear children. So the genotype of the parent should be IA, IO, IB, IO, or you can also write it like this, that is IB, I, and IA, I. That is also denoted like this. So this is the answer for the question. Okay? Chalo. Next, write 2 symptoms of Down syndrome and write the chromosome number in Down syndrome. So Down syndrome, my dear children, happens when the 21st chromosome which is there. So usually we have pair of chromosome. One we get from one parent, another we get from another parent. But from one parent we are getting 2 chromosomes. So what happens? There is an extra chromosome that is coming up over here. So you call this condition as trisomy. You call this condition as trisomy, my dear children, and these are not sex chromosomes, they are the other chromosomes, autosomes. So there is only one pair of sex chromosome, all the other chromosomes are known as what, my dear children? They are known as autosomes. They are known as autosomes, and this condition is known as trisomy, my dear children. So trisomy of the 21st chromosome happens up over here. So chromosome number, if you are asked, what will you write? So usually we have 44 autosomes, and then we have the sex chromosome which can be either XY or it can be XX. So 44 and then 45th and 46th chromosome, both of they are pair. So 46 number of chromosomes are there, but over here we have one extra autosome. So how do you write? 45A plus XX, that means it is a female who is having a Down syndrome, or it can be 44A XY, that is a male who is having what? Down syndrome. So your answer will look something like this. First of all, symptoms. Okay, symptoms are what? Individual will be short-statured, there will be round head, my dear children. The furrowed tongue will be there, tongue will be furrowed, and there will be partially opened mouth as well. Palm is broad and characteristic palm crease is also there. That means this line, no, it will be very much visible and will be different. Okay. Now, physical, psychomotors and mental development is retarded in them. The chromosome number in Down syndrome is trisomy of chromosome 21. The karyotype of Down syndrome individual is 45A plus XY. Okay, that is 47 male, or basically you can just write this. You don't need to write. Okay, or you can write, yeah, for is equals to 47 male, that is also fine. But you have to write the entire thing, otherwise teacher won't understand. Okay, or 45A XX, that is 47 female. There is one extra chromosome, right? Then, chalo. Next, define point mutation. Give an example for point mutation. What do you mean by point mutation? Where the mutation is happening only at one place. That is known as point mutation. Just give me a second. Let me see how many questions are left. Okay, from this chapter. Okay. Okay, so suppose you have GUG. This is a codon. Now over here, the U is replaced by A. You know what happens? This was coding for an amino acid that is known as glutamic acid. But now, since there is a replacement in A over here, it will be coding for valine. And this codon, my dear children, this is a part of multiple codons which are there, which is going to form hemoglobin chain, the beta globin chain. So now the beta globin chain would not be formed in a good manner. There will be quality issues in them, and that's how you get sickle cell anemia. Okay, that's how someone gets sickle cell anemia. So change in single base pair of DNA is known as point mutation. It is happening only at point. Classic example of mutation is sickle cell anemia, where the codon GAG, okay, sorry, it's not GUG which is getting, oh my god, okay. It is GAG which is replaced by GUG. How do you remember it? A comes before U. So A is replaced by U. Okay, A comes before U. So A was there and then it is replaced by U. Thank God I saw it now. Okay, now A is replaced by U, hence making the codon as GUG. Note that GAG codes for glutamic acid, whereas GUG codes for valine. And both of them are going to mess around the beta globin chain of hemoglobin. Now, what are the conclusions drawn by T.H. Morgan from crossing experiments in Drosophila with respect to linkage? So he got to understand one thing, that is when you talk about genes. When you talk about genes, okay, suppose this is a chromosome and this is another chromosome that we have over here. Cool. Now, these two genes are very close to each other. So, whatever type of recombination happens, when the chromosome part will be going from here to here, okay, from here to here, if it goes, my dear children, the new chromosome that you get will look something like this. But if you see the recombination percentage is less, the new combination percentage is less, because they are still together. Okay, there is another gene up over here, but they are still together, right? So these genes, I will call it linked genes. What will I call it? Since you cannot see it, I will say linked genes, my dear children. They are closely, or I will say they are tightly linked genes. So tightly or closely linked genes, they will be showing less amount of recombination. They will be showing less amount of recombination. But over here, if you talk about loosely linked genes, suppose this was not over here, this was not over here, this was somewhere over here. So when this particular thing will be cut and will be put over there. This particular part can easily go up over here and this can come here, right? There's a new combination that you're gonna get over here because it got interchanged. Now they are loosely linked. It can only happen when they are loosely linked. So loosely linked genes which are there, okay, loosely linked genes which are there, they are going to show high amount of recombination. Always remember, the amount of recombination is, the highest amount is 50%. So if you are said that 37.3% is being shown, that means it is showing lots of recombination. That means the genes are really apart. They are loosely linked, okay. And if it is said that only 1 point some percent is recombination is there, that means they are closely or tightly linked to each other. Cool? Chalo. Going ahead. So this is what you have to write. The higher the linkage, lesser is the recombination, and the lesser the linkage, higher is the recombination. In other words, genes which are tightly linked will show lesser recombination. The genes which are loosely linked will show higher recombination. Now, what is test cross? What is its significance? So basically, I have seen a plant and that looks tall, but I don't know its genotype. I don't know its genotype. Its genotype can be like this, capital T, or it can be small t. So what do I, sorry, not small t, it can be capital T, small t. Yo, where is it? It can be capital T, small t. In this way, basically this is dominating and here still will be tall. So how to confirm it, ma'am? Basically we can do a cross. With what? With a recessive parent. Now suppose there is a recessive parent over here and I am putting, just a second. I am gonna put recessive parent up over here, both the areas, okay? And I am going to put two parents. This is the heterozygous parent and this is the homozygous parent. Now over here, my dear children, small t are the gametes. Here the capital T are the gametes for the capital T parent. Now this is capital T, small t parent for their gamete looks like this, and here it looks like this. Now again I am going to make a box over here. Small t are the gametes of the recessive parent. Suppose this and this fuses, you get capital T, small t. This and this again capital T, small t. This and this capital T, small t. This and this capital T, small t. You see all of them are tall. That means the parent is what? Homozygous tall. That means the parent is what? Homozygous tall. It is pure tall, my dear children. Now let's see the condition over here, okay? Suppose this and this fuses, you get capital T, small t. This and this fuses, you get capital T, small t. This and this fuses, you get small t, small t. This and this fuses, you get small t, small t. So 50% of them will be tall and 50% of them will be dwarf. So in that condition, I understand that this is what? Heterozygous tall. This is heterozygous tall. So in that way, I can understand the plant that I am having is a pure line or it is not. That is the significance, and then I can use it for breeding program, right? So suppose I want tall plants, but I fused this with other plants, my dear children. There are chances that I can get dwarf plants as well. So that is the use of this. So how do you write it? It is a kind of back cross, okay? It is a kind of back cross where F1 plant is crossed with the recessive parent, or basically a plant is crossed with the recessive parent to understand its genotype. It helps in finding the F1 or plant is homozygous or heterozygous genotype, or just it helps in finding whether the plant is homozygous or heterozygous for one particular character, for one particular character. Then what is the significance? Basically, then I can use this pure line for crop breeding experiments or whatever I want. Chalo, next. What is phenylketonuria? Write the cause and the symptoms of phenylketonuria. So phenylketonuria is an autosomal recessive disorder. Okay, it is a recessive disorder. That means both the parents should be at least a carrier. That's when the child may have this particular disease. Okay. It will be only visible in the recessive condition. Both the alleles should be same. Now what happens in phenylketonuria, my dear children? Let me tell you that. So there phenylalanine, which is there in our body, it's an amino acid, okay? This phenylalanine is usually getting converted to something that is tyrosine with the help of an enzyme that is phenylalanine hydroxylase. That is what? Phenylalanine hydroxylase. Now, the gene coding for phenylalanine hydroxylase itself is mutated or deleted, which is why this conversion is not happening. And when this conversion is not happening, phenylalanine will be accumulating in our body in the form of something else that is known as phenylpyruvic acid. Phenylpyruvic acid. And now this will cause what, mental retardation and things like that, and it will be found in ketone, it is going to form ketone bodies in the ketone crystals in the urine as well. So a lot of symptoms are there which you should be knowing. So over here, it is autosomal disorder due to presence of recessive gene, okay, and you can go read this exactly what I said, okay. Now symptoms, accumulation of this in brain results in mental retardation. Phenylpyric acid and other derivatives are also excreted to the urine because of its pure absorption by the kidney. Infants have lighter skin, hair and eyes and everything. So they show hypopigmentation as well, because you know what, this tyrosine which should be produced is not being produced. This tyrosine helps in the production of melanin, and melanin is the one that gives us pigment. So in them, because of this, there is no pigment formation that is happening over here, which is why their skin and hair will be hypopigmented. Less pigmentation would be absorbed over there, okay. Now, T.H. Morgan selected fruit fly for his genetical experiment. Give any 3 reasons. What are chromosomal disorders? Give an example. So, shall I start with the first question? Yes. So, T.H. Morgan was working on chromosomal theory of inheritance, my dear children, where he took fruit flies. Why? Because the fruit flies had a very short lifespan. So if I want to see any result, I can see within 2 weeks, because that is a very short lifespan. And in them, the male and the female ones could be easily recognizable. So it would be easy to fuse them. Okay. Now the progenies, the number of progenies that they produced was way too much, and it was very easy for them to be grown in laboratory condition. Now what do you need? You don't need anything else, right? So those were the reasons why he chose the Drosophila melanogaster, or basically the fruit fly. So they could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks. A single mating could produce large number of offspring. They had a clear differentiation of sexes. Now, talking about chromosomal disorders, my dear children, what do you mean by chromosomal disorders? As in, when the number of chromosomes are being altered, or there is some structural differences in them, it will lead to chromosomal disorders. So disorder in human beings are caused due to absence or excess or abnormal arrangement of one or more chromosomes are known as chromosomal disorder. For example, we spoke about Down syndrome, right? In Down syndrome, there is an extra chromosome up there. Get it? And some cases there might be one less chromosome, such as Turner syndrome. In Turner syndrome, there is only one chromosome, one X chromosome that is coming from one parent. Another parent is not giving anything. You get it? So in Klinefelter syndrome, there is an extra X chromosome. So those are the things that are there. Cool? Have we actually completed that? So here we start with molecular basis of inheritance, which has again 13 marks weightage. And from this chapter, you will get at least one 2-marker question for sure. Okay, so we have to practice that, but before going ahead, let's already be in 1 hour 15 minutes, so I want you to take a quick break, drink some water, do some stretching and come back, and after that I will be starting off this session again and we will be completing all the other chapters. And if you have come this far, okay, if you have come this far, my dear children, you have to comment down that, ma'am, I did it, I completed half of the video. Okay, then we will meet again in the next half. Cool? Yes. Okay children, so going ahead with the next chapter, that is molecular basis of inheritance. So the total weightage is 13 marks, as I said, and over here there will be one 2-marker question for sure. So we have to practice certain 2-marker question. So which sequences of bases transcribed from DNA are found both in hnRNA and mRNA? What do you mean by hnRNA? Basically, you know, this is the DNA and this DNA has to form a transcript, that is RNA. Yes, that is known as transcription. So the first thing that the DNA forms during transcription is not mRNA but hnRNA. So what happens is that whatever non-coding sequences are over here in the DNA, that means the ones that do not code for any proteins, they would also be transcribed over here in the hnRNA. So it will look something like this. So they would have regions that is known as exons, which would be coding for something, some protein, okay, and they will be also having introns, my dear children. Now again, they will be having exons, and then in between they would be having introns that do not code for anything. Now you are asked, which sequence of bases are transcribed from DNA and are found both in hnRNA and mRNA. So this hnRNA will undergo some processing, such as splicing and all, okay. One such processing is splicing, where what do? We remove the introns. We do not want the non-coding regions, so we remove the introns and we join the exons. And now what you get is just so many exons together, my dear children, and this is what you call as mRNA. So what is common between hnRNA and mRNA? This sequence is, that is the exon, my dear children. Cool? Yes. And then further this mRNA will be undergoing the process of translation to form proteins. So it will look something like this. As I told you over here, these are introns and these things 1, 2, 3 are exons. So while splicing, you remove these guys and you join the other ones. So the sequence of bases transcribed from DNA that are found both in hnRNA and mRNA are exons, and exons are coding regions that remain in the mRNA after splicing, while introns are removed during the process of processing, my dear children. If you write this, you get 2 marks. Going ahead, mention the levels where gene expression can be regulated in eukaryotes. As in, for example, I have a gene, but I don't want this gene to get expressed. I don't want the protein that this particular gene is making. So at what all levels can I stop this, or basically can I regulate this? Now regulation can be both positive and negative. Okay, regulation can be both positive and negative. That means if I want a gene to make the protein, then also I would need regulation. If I don't want the gene to make proteins, then also I would be needing regulation. So in the case of eukaryotes, there are certain ways in which you can regulate. First is transcriptional level, as in when the DNA, as I said, will be forming the RNA. At this point of time, I can regulate. I would not allow the RNA polymerase to come and attach and give an RNA over here. Okay, you get it? That's how I can regulate it at one place, that is transcription level. And now processing level, as in I told you, it forms hnRNA first and then it has to form an mRNA. That's when it can make a protein further. So again, if I provide some hindrance over here, I do not allow this particular reaction to happen, I do not allow splicing to happen, or this is a very slow process, do you think it would be still able to make the mRNA? No. So again I can regulate at this particular level. Now what? Basically the whole thing that you see over here, it is happening inside the nucleus. It is happening inside the nucleus. Now, from the nucleus, this mRNA has to go outside to make the proteins. So this is known as transport of mRNA. So this transport also I can stop somehow. I can regulate over here as well. So this is again a level in which I can regulate the gene expression. I'm not letting this gene express itself, right? So as I said, it's mostly positive and negative, but I'm talking about the negative things because it becomes easy for you to understand that way. Okay, so transport of mRNA from the nucleus to the cytoplasm, that can be regulated. And now, suppose this guy went out. This mRNA went out. From here, the next process is translation. There also I can regulate. Okay, probably the tRNA which has to come, I would not allow that tRNA to come and bind over here or do something there. So again, in translational level also I can regulate the gene expression. So these are the 4 levels given in the NCERT which you have to definitely remember. Cool. Next, write a note on the role of sigma factor and the rho factor in transcription in prokaryotes. So what happens, my dear children? You know, again DNA is going to get transcribed, so it needs a enzyme, that is RNA polymerase. So RNA polymerase says, "Arey yaar, I cannot go alone. I cannot go alone for the process of transcription. You have to come together with me." Who is that? Who is he calling? So basically it is sigma factor that it calls first. So RNA polymerase, when it has to bind to a promoter region, from where basically it will come and bind, and then only it will initiate the process of transcription, my dear children. Here it calls the sigma factor as well for initiating the process of transcription. Now at the end, when it has to terminate, okay, so it reaches the terminator region, and again, so sigma factor comes over here and helps in initiation, and later they detach from each other. Then RNA polymerase does its own work. And now towards the end, for terminating the process, RNA polymerase need the next factor, that is rho factor, my dear children. So this guy will be coming and attaching to the RNA polymerase, and then only the whole termination process would be happening in the case of prokaryotes. So this is how it looks. So sigma factor binds to RNA polymerase, guiding to promoter region for transcription initiation. It detaches after initiation. So you see sigma factor and the RNA polymerase coming towards the promoter region and binding it over there. You get it? So it will be helping in initiation and later would be detaching. Now talking about rho factor, it facilitates termination by binding to RNA polymerase and unwinding the RNA DNA hybrid into rho independent termination. Basically the RNA and the DNA are together, right? So it will detach. It will make it detached, and basically the RNA would be now totally free. RNA will be free, RNA polymerase will be free, everyone will be free because of rho factor. Okay, you can remember rho as in cry, you know. So they were crying, now they are not crying because of this. Okay, because they are totally detached. It's totally fine. So they have ended the toxic thing. Okay. Either way, whatever way you want to remember, you can remember. But yes, sometimes you have to spice things up. That's how you can remember. Now, what are VNTRs? Mention the steps to detect VNTRs in identifying criminals in forensic investigation. So, VNTRs means variable number of tandem repeats. So, what happens is that me and you, my dear children, almost 99% of our DNA is same. Yes, yes. So, there are changes in that last 0. some percentage of the DNA, my dear children, and this is huge. So, this change basically occurs in 3 billion places that is around our body, and basically I would be having certain repeats in my body, certain sequences, base pairs which are repeated in my body, and this would be entirely different from what do you have. You also would be having repeated sequences. I also have repeated sequences. Now, these sequences are known as variable number of tandem repeats. Okay, there are variable number. I can have this repeats in 10 or 20. You can have your repeat in 30 or 40, whatever is there. Now, mention the steps how this particular thing can be used in identifying criminals. So, how can this be utilized? For example, there is a crime being done over there and there is blood sample. Now, I am the culprit and also there is one more guy over here. Okay, now both of our blood is taken and that blood is also taken. The sample blood which is over there, that is also taken. And now the DNA is sequenced. You have the DNA over there from the sample. This is the DNA from the sample. Now, what do? You basically make a complementary pair. You make a complementary pair of this particular basis and then you tag it. You tag it with some radioactive stuff. Okay, now this is known as a probe. This will be known as a probe. This is what? A complementary thing that I am creating. Now, over here, the culprit, that is me, my blood sample is taken. Again, my DNA is being taken from that, and my DNA is made to cut and then go to a gel electrophoresis chamber, and lot of things happen. Basically, they are separated. My DNA is being separated like this on the gel electrophoresis. Similarly, the friend who is there, their DNA is also separated. Cool. And now what is done? Basically, you take a nitrocellulose sheet, put it over there. All the content of the DNA would be over here in the nitrocellulose sheet. Now, basically, you make this probe run through the sheet, and then you will be able to find that this probe will be only attaching to its complementary pair. Yes, and that is where, my dear children, here, that is there over here. And with that, obviously, it is radioactive. Now, if you put it under UV, what will happen? This will be showing off its color. That means I am the culprit. I am actually the person who did the crime. So, this is how you do, criminal investigation with the help of VNTRs. So, I have tandem repeats, which you do not have, which is why this guy did not go and bind over there. This guy went and got bound over here itself, because these tandem repeats are very unique to my body. How do you write this? So, the DNA fragments that show very high degree of polymorphism are known as VNTRs, or variable number of tandem repeats. Now, what do? You do isolation of the DNA, digestion of the DNA by restriction endonuclease, it will cut it. Now, separation of the DNA by electrophoresis, then transferring of the separated DNA fragments to synthetic membrane, that is nitrocellulose sheet, my dear children, and then hybridization using labeled VNTR. I let this probe go, okay, and bind to any complementary pair. Now, detection of hybridization DNA fragments by autoradiography or UV, whatever is there, and that's how I find the criminal. It's interesting, right? So, yes. Now, write the differences between euchromatin and heterochromatin. Name the virus used in Hershey and Chase experiment. First thing, what do you mean by euchromatin and heterochromatin? You have to know that. So, we have a very long DNA, and this long DNA has to get fit inside of a very small nucleus. So, what do? You basically make the DNA coil around a group of proteins which is known as histone proteins. You have 8 histone proteins coming together. You call this octamer of histone. So, on this, the DNA basically wraps around twice, and then again there will be one more histone protein which is known as H1 histone. It will be going around that also, and then again there will be histone proteins, my dear children, almost 8 of them again, and this particular DNA will be wrapping around again. So, this is a bead-on string structure. So, basically you are making it more compact, and when you see it in an enlarged view, there will be so many beads on a string, and this structure you call it what? Chromatin, my dear children. This is what you call as chromatin. Now, basically the chromatin will be loosely packed or tightly packed. It can be compact or it can be loose. Suppose it is loose. At this point of time, when it is loose, when there is space between two, let's say histone proteins, my dear children, you will be calling this structure as euchromatin. Now, when it is tightly bound, you will call this structure as heterochromatin. Okay, euchromatin and heterochromatin. And I'll tell you one more thing. Since euchromatin is where the space and everything is there, what will happen when the DNA is in bound state like this only, here only transcription can happen. The RNA polymerase can come, bind and let the transcription happen only if it is in loosened state. So I'll say euchromatin is the one which helps in it is transcriptionally active. But in heterochromatin, everything is tightly packed. How do you expect transcription to happen over there? So transcription would not be happening over there. Cool? Now, again, there would be some more differences. The chromatin that are more densely packed and stains dark and transcriptionally inactive is known as heterochromatin. So they take up the stain really well. So they are going to stain really dark. But over here when you talk about euchromatin, they are not going to stain. They are going to have light stain, my dear children, and they would be transcriptionally really active. You see there are gaps. Over here there are no gaps. Okay. Now, the virus which was used in Hershey and Chase experiment for doing, so what was it, my dear children? It was bacteriophage, which you remember looked something like this. It looked like a spider, right? So this was the virus, and you call it bacteriophage because it is going to affect the bacteria. Don't think that it is a bacteria itself. No, it is going to infect the bacteria, which is why it is known as bacteriophage. And that was used in Hershey and Chase experiment. I hope this is also clear. Children, always have a visual thinking, okay? Visualize and learn. In that way you learn better, and especially when in the case of biology where there's too much of theory and stuff like that, you would not be able to remember it for a longer run if you're just by hearting stuff. So please don't do that. And I really hope I am helping you with this by understanding the concepts, by making you understand the concepts. I know this video might be going long, but I'm pretty sure everyone who is going to watch this video will be taking something back with them. So that is a use, and if you're liking it, do let me know in the comment section because that really motivates us a lot. Now, let's the goals of Human Genome Project. What are the goals of Human Genome Project, my dear children? Human Genome Project had multiple goals, so I'll write it over here. So over here, identify all the genes in the human genome. That obviously is a goal. Now determine the complete sequence of DNA in the human genome. First, identify all the genes. What genes are there? This gene codes for this and things like that. Now you have to have the complete sequence of base pairs also. What all base pairs are there in this particular genes and everything, the whole genome, okay? And after that, you have to store the information in the database, and then you would have to improve tools for efficient data analysis, and then transfer related information to other sectors and industries. Transfer this information which is there to other areas, okay, wherever it is needed, and address ethical, legal and social issues, that is known as LC, okay, arising from the genome studies. These were the goals of Human Genome Project. Much repeated question, so you should be knowing about it. Okay? Chalo. Next, mention the criteria for a molecule that acts as a genetic material. So if something wants to act as a genetic material, whether it be protein, whether it be DNA, or whether it be RNA, what is the first thing that you should be thinking about? That is stability. It should be highly stable. Suppose the DNA is not stable, it is undergoing changes here and there. We will also change like that. So do you want that? No. So it should be stable. It should be less reactive. It should not be so much reactive, right? So DNA becomes the best genetic material because it is not so reactive and it is highly stable, my dear children. Apart from that, what are other features that you should be remembering? See, it should be able to generate its replica. Obviously, when one cell divides into two, the genetic material also should be dividing into two. Otherwise, how would that particular cell perform? So it should be able to replicate properly. Now, it should be chemically and structurally very stable. And apart from that, it should provide the scope for slow changes if there are no changes at all happening. Okay, even if we say that it is stable, but still we expect little amount of changes in there. Why? Because if no changes is happening, no evolution would be happening. And if no evolution would be happening, my dear children, how would we survive? So evolution is also always for better. The organisms which are able to thrive better, they are able to survive. You get it? So there should be certain changes also. So it should allow certain scope for certain changes, small changes. Okay. Now it should be able to express itself in the form of Mendelian character. That is, whatever genes are there in that particular genetic material should be able to express itself. Otherwise, what's the use of having a genetic material at all? So these are the features of a molecule that acts as a genetic material, and all the features are being fully satisfied with the help of DNA. RNA is not chemically and structurally very much stable. So initially we had RNA as the first genetic material, my dear children, but then it was not stable. It was acting as an enzyme. It has an enzymatic activity also, RNA. So basically, obviously something that has an enzymatic activity or catalytic activity cannot be working as just a genetic material. Okay, so RNA is genetic material for some organism who evolved really fast. You know, viruses and all. Why are we not able to find cure for HIV/AIDS? Because this guy is just changing its genetic material here and there. So whatever we do, it changes of something into something else. So that is the thing with them. But it's a virus, and we are human beings, and we are much more complex. You get it? So, yes, and now we have reached the next chapter, that is evolution, my dear children. Evolution will give you 7 marks, and there will be 1, 2 and 3 marker question pakka coming from this particular chapter. So we should be knowing the answer to these questions as well. First question, potato tubers and sweet potato tubers are result of convergent evolution. Explain. So basically, my dear children, potato that you see, okay, potato that you see is basically nothing but the stem growth. This is a stem growth that is there. So it is a stem tuber. Okay, now talking about the sweet potato. Sweet potato, if we see, we have the stem, but the roots which are there, they would be storing nutrients over here. So it is a stem, it's a root modification, not a stem modification. So this is what? A root modification. Okay, so if you see, function of both of them is what? The same, that is what? Storage. And why are they storing? Because they are remaining in the same habitat where storage is very much required. So I'll say function is same, but structurally, do you think they have a common ancestor, or basically they have any structural similarity? No, because this is a stem tuber and that is a root tuber. So basically they might have converged to a point to have a common function. Why? Because both of them have nothing else to do but to store nutrients. So this is an example of convergent evolution, my dear children, where they are converging to have common function because of the habitat that they are living in. But they do not have a common ancestor, they do not have same structural features and stuff like that. Okay, now these organs which are there, you call these analogous organs. So convergent evolution is shown by analogous organs. How do you remember it? A, C, and the other one, homologous organ, but divergent evolution, HD. Okay, always remember. Now, so how do you write? Potato tubers are modified stem, sweet potato are modified roots. Okay, similar function. Both are adapted to storing nutrients, mainly starch, to support the plant, but independent evolution. These adaptations evolved separately in unrelated species to serve the same purpose, making them a case of what? Convergent evolution. So they evolved separately into serving a same purpose, that is having a same function, and that is why you call this what? Convergent evolution. Done? Chalo. Next, write a short note on Neanderthal man. What do you write about Neanderthal man, my dear children? Neanderthal man basically is like very recent form of man. I cannot say very recent form of man, but yes. So we have modern Homo sapiens, that is us, right? So just before us, okay, while we were evolving, at that point of time, someone else was also evolving a bit before us also. That is almost 1 lakh. I hope the zeros are enough, or did I write 10 lakh? No, okay. 1 lakh to 70,000 years ago is when they actually developed. That is Neanderthal man, my dear children. Just a second. I'm so sorry. Yes. So, I was saying 1 lakh to 70,000 years ago, they developed, my dear children. And what is very unique about them, their cranial capacity was way too much. It was almost around 1400 cc. It was around 1400 cc. Apart from that, what is a good thing about them? Basically, they were the ones who started protecting their body. They covered their body and they also buried their dead. So before that, the sapiens which were there, okay, sorry, not the sapiens, basically the human beings, the form of human beings which were there, they were not burying their dead and all. But Neanderthal man were the ones who buried their dead and also protected their body. So these are the features that you should be definitely writing about Neanderthal man. And I'll tell you one more thing. During this stage only, the Homo sapiens, somewhere around in the mid, the Homo sapiens also started evolving. Okay, now this happened in 2 separate areas. So yes, so these 3 points should be there. They evolved around 1 lakh to 70,000 years ago, and they had a cranial capacity of 1400. They buried their dead and protected their body as well. 3 points. Okay. Now, mention the evolutionary significance of the following organisms, shrews and lobfins. So, you know what? I'll talk about lobfin first. So, there was a fish, we thought it was extinct, but people who went on voyage and things like that found this fish, and it was known as coelacanth. And also this is the same thing that is known as lobfin. So it has very big fins, okay, and with this fins it was able to actually move on the land also. This was a fish, but it was able to move on the land. So this particular fish, my dear children, becomes an evolutionarily very significant organism, because it is assumed that from this organism only the modern day amphibians might have evolved. So it is like a connecting link. So the modern day amphibians, as in the frogs, salamanders, everyone might have evolved from them, that is lob fins. Now talking about shrews, so you know, basically first thing that we had is fishes, that is Pisces. From Pisces we had amphibians, that is frogs and all, those who could live both on land and water, and then reptiles, mostly they were living on the land, okay, some of them on the water and the water also. And then we had, from reptiles we had birds, that is aves, and also mammals. So shrews becomes the first form of mammals which were there. So they had this, what say, fossils which were looking similar to reptiles as well as mammal in a lot of sense. So they again became a connecting link between reptiles and mammals. So how do you write it? Shrews are most common, are among the earliest mammals and represent primitive ancestors of modern mammals, providing evidence of mammalian evolution from reptilian ancestors, my dear children. Okay, so that was the connecting link. Okay, shrews connecting link between reptiles as well as the mammals. Now talking about lobfins, they mark a critical step in vertebrate evolution, bridging the gap between fishes and the land dwelling animals. These animals are known as lobfins, evolved into the first amphibians that lived both on land and the water. They were the ancestors of the modern day frogs and the salamanders. So how do you write it? You just write it, lobfins basically evolved into the modern day amphibians, the first amphibians that lived on both land as well as water. And shrews are what are among the earliest, the primitive mammals which are there, and they share little characters from the reptiles and the mammals. So they are assumed as a link between the reptiles and the mammals. Okay, that's all. And now, what are analogous organs? I just told you what are analogous organs. So whenever you have the same function, but structurally they are not same, they do not have a common ancestor, these things are what? Analogous organs. For example, the most basic example is the wing of butterfly and the wing of bird. So if you see wing of butterfly, if you just go touch it, it will break. Do you think it is structurally same to the wing of the bird? If you do anything, it will still fly, my dear children, because it is thick, it has muscles, it has feathers, lot of things. But both of them, aren't they using the wings for flying? Aren't they using the wings for flying? So yes, the function is same, but the ancestor, structurally are they same? No. So whenever the functions are same, you will call this analogous organs, and this is a result of convergent evolution, my dear children. When we are both converging, because they are converging into the same habitat, which is why they have common functions over there. Now, the organs which are different in structure and origin but perform common functions are known as analogous organs, such as wings of birds, bat and the insects, the eye of octopus and mammals. The octopus also has eyes, we also have, mammals also have eye, but is it structurally same? Not at all. Both of them are using it for seeing things, but is it structurally same? No. Now flippers of penguins and dolphins, again, flippers in penguins, my dear children, they do like this and they walk. Over here in dolphins, they basically swim with the help of it. So obviously that is also different. Sorry. Flippers of penguins and dolphins, basically they are doing a common function. Okay, so I just thought of penguins walking. So penguins, when they get into water, with the same flippers, they would also be swimming, and over here in the case of dolphins also, they would be doing so. But dolphin is a mammal, penguin is a bird, my dear children. Do they have a common ancestor? No. Okay, they do not have a common ancestor. So sometimes what happens is that when a teacher talks a lot, especially during long classes, at this point of time, they also sometimes do not realize what are they talking about. That is what exactly happens with me at times. But whenever I spot these mistakes, I do help in the correction in the comment box itself. So please forgive me. But yes, this is the thing. So flippers of penguins and dolphins, my dear children, these are analogous organs. Why? Because this is an ave, a bird, and this is what? Mammal. And both of them are using the flippers for what? Swimming around. Cool, but they're structurally not same, and obviously ancestrally also they are not same. Now root modification in sweet potato and stem modification in potato is also an example, because both of them are functionally same. They are both used in storage, but structurally not at all. So these are analogous organs. I hope you remember it for a long run. Now another question, enumerate on convergent and divergent evolution with suitable examples in plants. So in plants, stem and root modification of sweet potato and what say the other potato, normal potato, is convergent evolution. Now if we talk about divergent evolution as in basically when they have a common ancestor and they are forming from the same unit, but functionally they are doing different functions, opposite of convergent evolution. For example, you have two plants, one is bougainvillea and the other one is cucurbita. So they have axillary bud, my dear children. In the case of bougainvillea, this axillary bud will be forming a thorn. It will be forming a thorn. But in the case of cucurbita, this will be forming a tendril. So over here, one is forming a thorn, other is forming a tendril. Both of them have different functions. But are they evolved from a common ancestor? Yes. Do they have the same structural features? Yes, they have both evolved from the axillary bud. So I will say they have diverged, they have moved differently because they are not living in the same habitat, they are living in a different habitat, but common ancestor is there and they also have similar structures internally. Okay, they have evolved from the similar structure, that is what you can say. So these examples you can give. So divergent evolution, the same structure developed along different direction due to adaptation to different needs is known as divergent evolution, and these structures are homologous organs, HD, thorn of bougainvillea and tendril of cucurbita. Same thing for convergent evolution. Okay, next. 3-marker question. Write the salient features of the following human ancestors. You have Dryopithecus, Ramapithecus, Australopithecus, Neanderthal, Homo habilis and Homo erectus. Now if you listen to me carefully, you will remember the whole evolution properly. So my dear children, we had, okay, before Australopithecus, we had someone, where is it? Just a second, okay? Okay, so before Australopithecus, we had, my dear children, Ramapithecus and Dryopithecus. So Dryopithecus, which were more ape-like, they were more ape-like, whereas when you talk about Ramapithecus, they were more man-like feature, they had more man-like features. So primates called Dryopithecus and Ramapithecus were existing. They were hairy and walked like gorillas and chimpanzees. So this is one feature, they walked like what? Gorillas and chimpanzees. Ramapithecus were more man-like, while Dryopithecus was more ape-like. Okay, so you can write both these features for Ramapithecus and Dryopithecus. Now, Ramapithecus basically, which was more man-like, they evolved into Australopithecus. They evolved into Australopithecus almost 3 to 4 million years ago. Now, they also had man-like features. Again, they had the modern human-like features, at least some, okay. So there is something that you can write about Australopithecus is that they didn't go on hunt. They basically ate fruits, okay. They basically ate fruits and didn't eat meat. Or basically you can write they ate fruits and they were tool makers for Australopithecus as well. Because in your book there is no proper segregation, but actual thing I'll tell you. So in Australopithecus it is written that they were like human-like, okay. Australopithecus were more human-like and they started making tools and everything. They started making tools. So you can write that point in Australopithecus. Okay, you can remove from Homo habilis and you can put it in that point. Now, talking about Homo habilis. So, they were known as Homo habilis because they had pakka first hominid features, that is pure human-like features were there, and they had 650 to 800 cc of brain. The cranial capacity of brain was 650 to 800 cc, and they were the ones, they didn't eat meat, they ate fruit, my dear children. They ate fruit. Now obviously when they evolved from them, they were also like tool makers only. So in your book there is no clear distinction, which is why I told you it might be confusing for you guys. So for the first thing, what you can do is that Ramapithecus, you can write they were more man-like and everything was there. They were hairy, still walked like gorillas and chimpanzees. But yes, they were more man-like than Dryopithecus. Now talking about Australopithecus, which came from them, okay, they were living in Tanzania and all, okay, that you can write. They were living in Tanzania and they were mostly tool makers. That's all, 2 points. And now write about Homo habilis, that they are the first hominid, okay, and had 650 to 800 cc of brain. They ate fruit, they didn't eat meat at all, and they were tool makers. They were what? Tool makers, my dear children. Now from Homo habilis, you got Homo erectus, which had 900 cc of brain. You see the brain capacity, the cranial capacity is increasing. They started eating meat. And they were also known as Java man. Why? Because the Homo erectus, some of them went to China and all, and China, there is a region that is Java, which is why you call it as Java man as well. So 900 cc was their brain, ate meat, and they were Java man. Now from Homo erectus, two things came upon. One is Homo neanderthals, that is Neanderthal man, my dear children. About 1 lakh to 70,000 years ago they came. They had 1400 cc brain, and they protected their body and buried their dead also. So if you see, the brain capacity has always been increasing, and during this point of time, between this point of time, someone else was also getting developed, that is modern Homo sapiens, that is almost around 75,000 to 10,000. It is not, these many zeros are not there. Okay, 75,000 to 10,000 years ago, my dear children, during this particular time. Okay, during this particular, in between this particular time, Homo sapiens were also evolving. That is, and they are known as the modern day humans. They are known as the modern day humans. So first thing, you have Ramapithecus Rama, that it's man-like. Okay, so you can remember like that. And now Australopithecus, from Australopithecus Yel Habibi Homo habilis. Okay, the first hominid, 650 to 800 cc brain, ate fruits, didn't eat meat at all, and they were tool-makers. Now Homo erectus, again, if you talk about the 900 cc brain, ate meat, and they were Java men. They spread through different areas. Okay, so just China and all. Now from Homo erectus, you got two things. One is Homo neanderthals, that is about 1 lakh to 70,000 years ago they came, 1400 cc brain, and they protected their body, buried their dead. Talking about Homo sapiens, they are the modern day humans, and they evolved around 75,000 to 10,000 years ago. That's all. And with that, you complete this question as well. Now, write a detailed note on adaptive radiation. What do you mean by adaptive radiation? For example, I am Darwin, and no, I'm not Darwin. Darwin came to this particular area, that is Galapagos Island. Okay. Now, what did he find? That there were certain finches. But then when he moved to this area, he could find the same birds, but with different beaks, different beak structure. And there when he went, there he found another finch with different beak type. So probably it might have worked like this. That is, the finches were there, and the finches, among them, they had certain mutations. Some of them had mutations, which is why their beak was different. And now suppose they were in an island, so obviously they would be moving around certain areas. So certain guys from this gang went to that side, and they got to know that they are able to eat food there. They are able to eat insects with the help of this beak that they are having. So basically they evolved into other form of finches over there. Now some of them, again with some mutation, went up there, and they found that I am able to evolve much over here. I am able to eat grass and all with this beak that I am having. So basically from this seed eating type of finches, they evolved into insectivorous finches, they evolved into vegetative finches, and so many different type of finches. So over here, this is adaptive radiation. They are radiating and they are getting adapted over there. So if you see, my dear children, what type of evolution do they show? They show divergent evolution. They are diverging. Okay, they are diverging. They had a common ancestor. They are diverging because they have different habitats to stay. Get it? This is an example of divergent evolution. But you also have to remember one very important thing. Suppose there was one more example given in your NCERT, that is about the Australian island. Okay, so in Australia, what happened? You had two things. One is placental mammals, and the other one is Australian marsupials. Australian marsupials and placental mammals. Both of them were staying in the middle, and they radiated to different different areas. So from Australian marsupial group also one guy went there. Okay, from placental mammals group also one guy went there. So both of them had a common feature. Even though they evolved from different organisms, we are different organisms, still over there they are doing a common function. So this type of evolution is what? Where they are evolving into having a same function, okay, because they are in the same habitat. This is convergent evolution. So when you have two groups of organisms coming at one point, you call this convergent. When you have one group of organism going, if you talk personally, if you just talk about placental mammals, yes, they are showing divergent evolution. They are moving to different areas, evolving differently. But when you talk about Australian marsupials also, they are also there, and they are also moving to different areas, and now both of them looking the same. So basically that is convergent evolution. So you have to write little about all these things. So the evolution of different species in a given geographical area, starting from its original character and radiating to other geographical area, is known as adaptive radiation. That is finches in Galapagos Island. During Darwin's journey to Galapagos Island, he observed small black birds called Darwin's finches. The finches were diverse with their food habits. Okay, from originally seed-eating features to many other forms with altered beaks. Okay, now adaptive radiation in Australian marsupials. A number of marsupials, each different from others, evolved from an ancestral stock but all within the Australian continent. For example, Tasmanian wolf, tiger cat, marsupial rat, kangaroo and everyone. Now talking about placental mammals, they also did divergent evolution. Okay, in Australia, adaptive radiation is exhibited where placental mammals are evolved into varieties, each of which appears to be similar to corresponding marsupials. So the marsupials, they were kind of similar to marsupials. This is an example of what? When two of them are talked together, you say it is convergent evolution. Okay, so over here, Tasmanian wolf. Okay, Tasmanian wolf, that is from here, and the placental wolf, that is from here, both had the same features because they were obviously coming to a same point. You get it? So please write all these examples, not in this much detail, but just the points. Okay, so first the definition and then go ahead with everything. Now, with the help of a neat label diagram explains Stanley Miller's experiment in support of chemical evolution. So what does chemical evolution say? It says that basically the inorganic things that are there, for example, methane, ammonia, hydrogen, all of them, they came together under reducing environment when the oxygen was not there and when temperature was very high to form organic components such as amino acids and stuff like that. And later these amino acids probably grouped together to form proteins, and some nucleic acids probably might have grouped together to form RNA and things like that. That is how evolution has happened. This is chemical evolution, my dear children, and to prove that, Stanley Miller did an experiment where he had a setup like this. You have to make this diagram. Okay, so over here from the boiling water, what do you get? You get water vapor. And the electrodes over here provides high temperature, that is around 800 degrees Celsius. You have to remember 800 degrees Celsius. And the gases were also provided: methane, ammonia, H2 and basically H2 gas. So this is a reducing environment because it's a closed chamber, there is no oxygen. At this condition, what do you find? Basically, you find certain liquid trapped over here, and this liquid had organic matter, that is amino acids. And with this, he could prove that yes, from inorganic things you get organic stuff. And probably this is how evolution might have happened. Okay, so this is a very important thing. Draw this. So this whole thing that I just explained is in written format. You can read it to it whenever you want. The PDF will be available. My dear children, you have to join the WhatsApp group for getting the PDF. For all the children who are asking for the PDF, it's there in the WhatsApp group. Please join it. Going ahead, state Hardy-Weinberg principle. Explain any 4 factors affecting Hardy-Weinberg principle. So Hardy-Weinberg principle says that the frequency of alleles in a population would be same generation after generation. That is, for example, I have 2 alleles, that is capital T and small t. So the frequency of these alleles, my dear children, he said it will be same generation after generation. Or there will be 50% of this and there will be 50% of this always. But is that true, my dear children? No. And how did he represent it? Basically, he said frequency of allele T can be calculated with the help of P and frequency of small t can be calculated with Q. This is how he said. Now, was it actually happening in this way? No. Because suppose some other group of organisms came in who were tall. So in that case, obviously, the capital T allele will be more. The capital T allele will be more. The frequency is changing over here. What do you call this feature as? This is gene flow. What caused this difference, my dear children? Because of gene flow. The gene is flowing. Some people are coming over here. Okay, bringing their genes obviously. So there will be gene flow. This can change the Hardy-Weinberg principle. Now suppose, accidentally all the tall people were killed. Accidentally the tall people were killed. So what will happen? Basically, you are left with small t only. Now that is known as genetic drift, where there is a frequency change because of any random chance. This is known as, I don't know what I am writing. Basically, it is known as genetic drift. There is a sudden change in the population. This I can explain better with another example. So there were certain beetles. The white ones, which were very much more in amount, and the green ones. So the frequency of these alleles are less, but the frequency of this is more. Now one day an elephant came and stamped off all in this. What happened? By chance, all of them got killed. Now whose frequency is more, my dear children? It was the green one who was more. So this is the example of genetic drift. Again, suppose this is a population that is there. Now all of a sudden what happened? Basically some mutation happened such that this turned into yellow, yellow, yellow. Again, isn't the frequency changing? Yes. So mutation can also be a reason. Now these organisms which are there, they're sexually reproducing. Suppose in sexual reproduction you have recombination. If you know the chromosomes comes together, they recombine, they have new things coming up. So again, genetic recombination happening during sexual reproduction is also one big reason. How many reasons do you want? So basically these are the factors which would be affecting the Hardy-Weinberg law. So the principle says that allele frequency in a population are stable and remain constant from generation to generation. Now gene flow, the sum total of genes occurring in sexually reproducing population in a geographical area at a given time is known as gene pool. Okay, the transfer of genes between the population of different gene pool which differs genetically but can interbreed is known as gene flow. So there will be gene flow that will be obviously leading to disturbance in the frequency. Now genetic drift, it is a random change in gene frequency in a small population purely by chance. Now mutation what spontaneous changes and permanent changes that is happening in the genetic makeup of an individual. Going ahead, we have genetic recombination. It is a process of formation during gametogenesis result in the formation of new combination of character, again disturbing. Now natural selection is also there, one that I didn't explain. So natural selection, my dear children, is basically, suppose over here there were white ones and these white ones could be easily spotted, but the black ones, oh my god, okay, where was I? Okay, the black ones could not be spotted that easily, so they survived. So this is nature's way of selecting things. So these frequency, again, they will be lost since they would be spotted easily by the predator. You get it? So that is natural selection. It is the process of selection of better adapted individuals with useful variation. So fittest or the better one will be surviving and the other one would not be. So again, changing the genetic frequency overall. So I hope you got that thing as well. And with this, we complete that chapter also and we get into the next chapter, that is human health and diseases, carrying 11 marks, my dear children. Why I have not written how many questions are going to come? So I think there is going to be a short 2-marker question and a 3-marker question. Just a second. Let me see and then let me tell you. Yes. So you would be having short 2-marker and 3-marker from this particular chapter. So where am I? I'm here. Okay, draw the structure of an antibody molecule. My dear children, this is a very important picture. So in antibody molecule, you have to mention 2 heavy chains. This is one heavy chain, this is another heavy chain. You have to mention 2 light chains, okay. Now over here, please remember, in both the chains, you would have a region which is known as variable region. So this region, okay, I can show it. This region that you see over here, these both regions are known as variable regions, whereas from here to here and here, it is constant region. It will be constant in all the antibodies, but this will be different in all the different type of antibodies. Now if you see, there are disulfide bonds between this, okay, between the chains, heavy chains, you have multiple of them, but over here between light and heavy, you just have one, okay. Now apart from that, these chains, my dear children, they are polypeptide chains. That means they are made of amino acids. An amino acid has 2 ends. One is an amino end and the other one is carboxylic end. So over here you have the carboxylic end. Okay, suppose the amino acids are changed together. One side will be having the carboxylic end and the other side will be having the amino end as well. Now the most important thing, this region, that is antigen binding site, which is there in the variable region, my dear children, this is where the antigen will be coming and binding. You have to make this diagram properly and label it, okay. Now, list any 2 methods of HIV transmission. How can HIV be transmitted? With the help of sexual contact. Apart from that, what is the other thing? Basically, if the blood needles, okay, the needles, syringes and everything is being exchanged between 2 organisms or basically 2 human beings, one who is having HIV and the other one who is not. Basically, this is how the HIV transmission can be happening. So, sexual contact with infected person, transfusion of contaminated blood and the blood products, sharing infected needles, and from infected mother to the fetus through placenta. Through placenta, usually the blood does not get mixed, but somehow if it gets mixed, then obviously child will get it. Organ transplantation from an infected donor to what? Recipient. That is also one big way. Okay. Going ahead, name 3-marker question, okay. Name of the drug, its source and nature is given below. Find A, B and C. So this is a model question paper question. You should be knowing it. So my dear children, name of the drug, the source of the plant is Papaver somniferum and it is a depressant. So my dear children, Papaver somniferum is opium poppy, that plant, and opium poppy has just one depressant and that depressant's name is heroin. What is its name? Its name is heroin. And the other drug obtained from this is morphine. So morphine's chemically other version is known as heroin, my dear children, and heroin acts as a depressant. Morphine is a painkiller and a sedative, not a depressant. Now talking about cannabinoids. Cannabinoids are produced from what? And they have effect on the cardiovascular system, that is basically your heart and everything. So cannabinoids are the only one that affect on cardiovascular system. Rest, this and this, Okay, that is opioids and cocaine, basically coca alkaloids. Both of them have effect on CNS, central nervous system, your brain and your spinal cord and everything. So this is the only one having effect on your cardiovascular system, and it is obtained from cannabis sativa. Cannabis sativa. Okay, now talking about cocaine, cocaine is obtained from erythroxylum coca, and as I told you, where does it affect? It affects your CNS, central nervous system. So papaver somniferum, opioids, my dear children, opioids, it affects your CNS and also your gastrointestinal tract. So this is CNS and gastrointestinal tract, this is cardiovascular system, and this is just CNS. It causes hallucination. This guy causes hallucination. You have to remember it. And this is the one that messes up with dopamine. Yes, going ahead. Okay, come this side, come this side, come this side. You have to come this side. It's not coming. Okay, then I have to go that side. Now, describe any three properties of cancerous cells, my dear children. Cancer cell, basically, okay, so normal cell, when it divides, okay, and when some other cell is also dividing, when they come in contact, they stop division. This is known as contact inhibition. But in the case of cancer cell, they lose this property. So I'll write loss of contact inhibition, my dear children. Apart from that, there are certain other features also. They show metastasis. What do you mean by that? Basically, the cells over here, if they are dividing over here, they would be moving around different body parts and will be spreading over there also. So that is there. Now, uncontrolled growth, they divide rapidly without responding to normal regulatory signals. So they do not respond to normal regulatory signals in our body. So in our signals, they say that they do not have to divide, but still it keeps on dividing. So uncontrolled growth, metastasis, lot of contact loss, not lot of, basically loss of contact inhibition is also one character of cancerous cells. Cool. Now, name the pathogen, vector and the symptom of malaria. A very easy question. Pathogen is what? Malaria is caused by Plasmodium. Okay, it can be falciparum, it can be vivax, it will be malaria, it will be either thing. Pathogen. Now, vector. Who is the one that carries it? It is Anopheles mosquito. What Anopheles mosquito? The female ones can only come and drink the blood. Okay, the male ones have the plant sap. So it is female Anopheles mosquito. Now, what is symptom of malaria? You get fevers, you get chills at times. So those are the symptoms of malaria. So pathogen is Plasmodium, P. vivax, P. malaria, and P. falciparum. Vector is by biting of infected female Anopheles mosquito, and symptom is high fever and chill that occur on very alternate days. And now, next question, differentiate between active immunity and passive immunity. My dear children, listen to me carefully. So for example, I have some foreign particle in my body. Okay, now my body is producing antibodies against it. So that means my body is actively participating. This is active immunity. So to ignite active immunity in my body, what do? People generally do vaccination. Why vaccination, my dear children? In vaccination, they provide dead and basically not dead, weakened pathogens, okay, that cannot cause any trouble to us. They just provide it to us so that our body knows that, hey, we have to produce antibodies. So it produces antibodies. But what happens the first time it produces antibodies, it cannot basically deal with it, which is why we get sick a bit, okay. But the next time the same pathogen comes in, we will be actively responding to it by producing lots and lots of antibodies, because our body would be producing certain memory cells. Now, on the other hand, what do you mean by passive immunity? Well, basically, I am not letting my body produce antibodies. I am not waiting for my body to produce antibodies. Instead, I am providing antibodies to my body. For example, a baby is there. When the baby is born, it does not have too many antibodies to fight against it. So what does the mother do? Mother feeds them with milk. And what does the milk have? Milk have antibody A, IgA. Now this is ready-made thing that I am giving to the baby. Why? Because the baby cannot produce it. Now that is passive immunity. Now you get a snake bite. Doctor gives you antivenom. What does this antivenom contain? It contains a preformed antibody, because you cannot wait for your body to produce antibody against it as of now. So this is passive immunity, my dear children. This is the difference. So active immunity, it will be staying throughout, but passive immunity, it will be temporary. Until you have those antibodies that I have given you, you would be safe from that. You know, it is temporary, that is there. Now there are much more differences. You can see it over here. So active immunity, the immunity developed due to injection or injection of exposure to infection. What is happening with me? So whenever I talk very fast, that's when I go blah 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 blah. injection or exposure to infection. Now, the immunity developed due to ready-made antibodies, that is this. Now, this is slow, it takes time to give full effective response, as I said, obviously. Now, it shows immediate response because you are giving antibodies, right, ready-made thing. Now, it provides lifelong immunity, but this provides temporary immunity. Now, the memory cells are produced for long-term immunity, yes. So, whenever something happens, something comes into my body will be producing memory cells. But over here, memory cells are not being produced because you are giving antibodies. No, then there is no need of it. You get it? Now, define allergy. What is allergy? It is what exaggerated, hyper-exaggerated response to something in our body. That is allergy, my dear children. And in allergy, antibody E is the one that plays a major role. So, antibody E go and settles on some cells which is known as mast cells, my dear children. They will be coming and standing like this, and they will capture antigens. Now, this mast cells which are there will produce histamine and things like the serotonin, which causes inflammation in our body and stuff. Now, mention the types of carcinogen with the example of each. Carcinogens are cancer causing things. So, what are there are physical, biological and chemical, a lot of things. So, let's see, physical carcinogen, that means ionizing radiation such as X-rays and gamma rays, and non-ionizing radiation such as UV radiations, my dear children, they can fall on our DNA and brings out mutation in there. Now, chemical carcinogen, you have tobacco smoke, industrial chemicals and things like that, again causing lung cancer and stuff. Biological carcinogen, as in the viruses causing cancer, they are known as oncogenic viruses with oncogenes. Okay, there are viruses which transfer their genes into us, and they are the ability to cause cancer in our body. Apart from that, there are several genes called cellular oncogenes. You know, in our body, there are certain genes which are doing some function, doing some random function. But if at all they are mutated, they will be turning into cancerous genes. So, they are known as proto-oncogenes. They are proto-oncogenes. So, until and unless they are not mutated, they do fine work. But if they are mutated, my dear children, they will be turning into what? Cancerous cells. So, they have been identified in normal cells, which when activated, obviously causes cancer. Okay? Okay. So, that is about this thing, and there is something that I want to tell you, but you have to wait for it. Okay? Just give me a second. Okay. So, we are done with that, and the thing that I wanted to tell you is here, my dear children. So, basically, I was talking about something really important. So, you are PU2 aspirants, obviously you are watching this video. Now, if you are someone who is thinking that you should be giving KCET this year, that is coming on April, my dear children, you have two options to prepare really well for KCET. One is Abhyas KCET test series, which is completely online, you don't have to go anywhere. One is Lakshya KCET test series, where you would be getting a chance to study with the teachers over here, and that will be a 21-day residential course or a day scholar option. If you are in Bangalore, you can come and attend, you can go, otherwise you can stay also here. So, what do you get over here is that there are 35 full syllabus and chapter wise tests in Abhyas KCET test series. There will be video solution, there will be what's analysis of past 5-year paper, and apart from that, if we talk about Lakshya KCET crash course, there will be daily practice test, there will be one-on-one doubt session, there will be study hours, focus study hours, okay, PYQ practice will be there, full syllabus would be covered. And if you have something else to ask to us, you can actually call in this number. Okay, so basically if you call this number, you can enroll to our course which is there. And if we talk about Abhyas KCET test series, my dear children, its price is 1,499. With this price, you will also get access to PU2 test series, that is for PU2. PU2 Success Blueprint test series is also available here. So, obviously, you have to take an informed decision, go through everything really well, take an informed decision, and if you need our help in doing so, do let us know in the comment section. So, we would be here helping you out. Okay, so yes, you can think about it, and now let's go ahead. So, over here, the next chapter that we have is Microbes in Human Welfare. It is a 7-marker chapter, it gives you 7 marks, and here both 2-marker and 3-marker or neither of them can come. Either both 2 and 3 or neither of them would be there. Either directly there will be a 5-marker question. So, my dear children, you should be knowing the probable number of questions, right? So, first is biofertilizers, and you have to mention significance. So, you know, when we use fertilizers, those can be chemicals, and these chemicals obviously have adverse effect on the soil quality and this and that. So, why not use organisms that can trap nitrogen, that can trap phosphorus for us? Yes, so those organisms are known as biofertilizers, my dear children. You might have always heard of rhizobium. So, rhizobium is a bacteria which is there, and rhizobium, it basically captures nitrogen which is there to this and provides it to the plant. So, what does the plant need apart from nitrogen? Obviously, there are a lot of other things, but as of now, let's assume that it needs nitrogen. So that is a big thing. So that is the significance. Biofertilizers and are organisms that enrich the nutrient quality of the soil. The main sources of biofertilizers are bacteria, fungi and cyanobacteria, my dear children. Now, nutrient availability is the significance. It increases the availability of nitrogen, that is done by rhizobium, azotobacter and things like that, and also phosphorus. So, there is a fungi in mycorrhiza. Mycorrhiza is a symbiotic association where that is association between plant and the fungus. The fungus will be going inside the soil and providing phosphorus to it. So, this is again a biofertilizer that is there. And rhizobium, azotobacter, they provide nitrogen to the soil. So what does the soil need, not the soil, the plant need apart from this? Now you have defined BOD and mentioned the commercial importance of Monascus purpureus. What do you mean by BOD? BOD is biochemical oxygen demand. Okay, that is demand for oxygen. So what happens, my dear children, during sewage treatment, you know what? For treatment, the primary effluent or basically whatever has to be treated is passed into a tank which is known as aeration tank, where you provide air and all. So in this aeration tank, who will be growing? There will be aerobic bacteria, those are the ones who grow over here. My letters are jumbling, my words are jumbling. Okay, these are the aerobic bacteria which would be growing over here and feeding on the organic matter up in this particular thing which has to get treated. Now, whenever this organic matter's amount will be reduced, that's when we say that the water is clear now. So, the aerobic bacteria will be feeding on it, and the aerobic bacteria will obviously need what, my dear children? Oxygen. They are aerobic, they need oxygen. So, basically, at this point of time when the organic matter is way too much, the aerobic bacteria will say that I need more oxygen to break things down. So, the amount of BOD, biochemical oxygen demand, will be way too much as well. Now, suppose the organic matter is less. That means the aerobic bacteria will say, I don't need much amount of oxygen for breakdown. Hence, what will happen? The BOD which is there, that will also go less. So, defined BOD, biochemical oxygen demand, is there. The amount of oxygen required by the aerobic bacteria in 1 liter of water, that is BOD, my dear children. Okay, and if the organic matter reduces, that means BOD will also reduce. So, BOD, the measure of BOD is the measure of pollutants. If the BOD is way too much, that means pollution is also way too much. Okay. Now, if you are asked about importance of Monascus purpureus, it is the one that produces something that is known as statin. Now, what is statin, my dear children? Statin is something that will stabilize the blood cholesterol level. It is a blood cholesterol lowering agent. It lowers the amount of blood cholesterol in the body. So, this is one very important thing that you have to definitely remember. Okay. So, BOD refers to the amount of oxygen required to oxidize total organic matter by bacteria present in 1 liter of water. Now, about Monascus purpureus, it produces statin, which acts as a blood cholesterol lowering agent. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So, the one that is responsible for synthesis of cholesterol, it competitively inhibits, as in it says that I am going to basically inhibit you, I am not going to make you, produce or synthesize enough amount of cholesterol. That is how it works. Okay, so this is again a 2-marker question. Now we have a 3-marker question. Complete the below given tabular column with appropriate answers. So, name of the microbe, name of the product and uses is given. What is the name of the microbe over here, my dear children, which produces lactic acid? It is lactic acid bacteria. And what is the use? Use is to convert milk into curd, that is the use. Okay, or basically to enrich the curd with vitamin B12, that is also use. Now you have methanogens. What is the product you get from methanogens? You get the product that is basically methane. Okay, the name is methane, H2S, CO2. Basically, the use is it can be used as biogas. Okay, it can be used in your kitchen, it can be used for electricity, things like that. Now, dash, and it is there for treatment of bacterial diseases. So, for treatment of bacterial diseases, you need an antibiotic. So, what could be that antibiotic given in NCERT? It is penicillin, my dear children. This is penicillin. Name of the product is penicillin. Who produces it? What is the name of the microbe? It is Penicillium notatum, which is a fungus. A fungus produces antibiotic to kill the bacteria. So, Lactobacillus is a lactic acid bacteria. It produces lactic acid, coagulate and partially digest the milk protein. That is the use. Okay, into forming the curd. You can just write milk to curd. Now, methanogens produce methane used as biogas. Over here, Penicillium notatum produces penicillin. Penicillium notatum produces penicillin, and it is there for treatment of bacterial disease. Done? Cool. Now, explain the role of microbes in industrial products. So, Acetobacter aceti produces acetic acid, Clostridium butylicum produces butyric acid, Aspergillus niger produces citric acid. These are the uses that you need to write. So, Aspergillus niger citric acid, Acetobacter aceti acetic acid, and Clostridium butylicum butyric acid, Lactobacillus lactic acid that converts milk into curd, yeast which is there, it produces ethanol that would help in the formation of brewery and things like that. Penicillium notatum produce penicillin. You are asked about industrial products. Yeah. Streptococcus produces streptokinase. What is streptokinase? It is a clot buster. It busts the clot, so there won't be any clot in your body. Okay. Apart from that, Trichoderma polysporum, cyclosporin A. Okay, cyclosporin A is what immunosuppressant. It suppresses the immune system. So when your body is like, I want to fight all those things, it basically suppresses it by producing cyclosporin A. Now, Monascus purpureus, it produces statin, which stabilizes the blood cholesterol level. Cool? Okay. Now, next thing, explain the role or role of microbes in household food products. So, household as in lactic acid bacteria. Over here, as I said, it converts milk to curd. It increases vitamin B12. Now, baker's yeast is there, that is Saccharomyces cerevisiae again. It is used to make the bread. Toddy, which is a traditional drink, it is again used, is made from the fermentation of sap from the palms. And the large holes in the Swiss cheese. In the Swiss cheese, the one that Jerry used to take away from Tom, my dear children. Not from Tom, but he used to basically go for that only. It was Swiss cheese, and in that very big holes are there. It is because there is a bacteria which produces CO2. So that bacteria's name is Propionibacterium shermani. It's going like, it's shermying. How do I say in English? I don't know. Now it is getting shy. Now this, the Roquefort cheese is ripened by specific Penicillium roquefort, that is a fungus. It is not given in your NCERT, but yes, it is a fungus that is going to produce it. Okay, so you have to remember all this thing. It is not producing it, basically ripening it. It ripens the cheese. Okay. Now, explain the role of three organisms as biocontrol agents. Okay, the separated bands of DNA are cut out. What is the significance of BOD? Give an example of fungus found in mycorrhiza. Okay. So basically, I'll start from the last question. Okay. Mycorrhiza is what? Association of plant and fungus. Now, the fungus which is usually forming the mycorrhiza belongs to a genus that is Glomus, not Phlomis. Oh my god, I don't know. What I want to write, what I want to say is not coming out as such. Okay, so basically it is Glomus, not Phlomis. It is Glomus, my dear children, because it belongs to this particular genus. Now, what is the significance of BOD? I told you, BOD acts as an indicator of pollution. If there is high BOD, that means there is more organic matter, which is why and if there is more organic matter, obviously more pollution as well. Now, the separated bands of DNA are cut out. Okay, they are cut out with the help of elution. Are you asking about the process? If you are so, then it is known as elution. The DNA bands which are separated, my dear children, it is cut out from the gel electrophoresis with the help of elution. We will see the answer also. Now, explain the role of three organisms or biocontrol agents. Now, biocontrol agents are the one that controls the pest in there. You know pest usually is there for damaging crops and stuff like that. So, we use pesticide, but we do not want to use pesticide. We want to use biocontrol agent which could be easily killing them. So, first example of biocontrol agent is, as I remember, is Bacillus thuringiensis. It is a bacteria that produces Bt toxin. Okay. Now, when the insect, they would be feeding on this particular bacteria, the toxin gets active in their gut and it will be basically killing the insect. So, that is one thing. Second thing is ladybird. Ladybird which is there, my dear children, it will be feeding on the aphids. Aphids is a pest. Now, third thing, you have dragonfly. Dragonfly feeds on mosquito larvae. 3 biocontrol agents. Okay. So, the ladybird and dragonfly are used to get aphids and mosquito. Bacillus thuringiensis, as I told, it will be basically used to control butterfly caterpillars. So, dried spores of this is been put with water and been sprayed. Okay, so that so that in the gut of the larvae the toxin is released and the larvae gets killed. Now, Trichoderma, free living fungus used to control several plants. So, Trichoderma, which produces cyclosporin A. It is also a fungus that stays in the soil. Okay, so there it will be fighting against other fungus. It is also fungus, but it will fight against other fungus. Now, baculoviruses are also there, my dear children. They basically attack insects and arthropods. That is there, baculoviruses, and they belong to usually nucleopolyhedroviruses. Now, all this thing is there. Apart from that, BOD is a measure of organic matter present in the water and also an indicator of the pollution, and Glomus is the thing that I was talking about. Yes, so the question was this. I don't know from where you got this, so I'll just cut this off. There is no question like that's what I was thinking. How did this came over here? Okay, got it. Now we have biotechnology and its application, which is a 6-marker chapter, my dear children, and you either get a 2-marker question from them or you get a 3-marker question from them. 2-marker and 3-marker, both of them would be there. Either 2 and 3-marker will be there, or both of them would not be there at all. Okay, so ELISA is one of the methods of molecular diagnosis. What is the principle of this technique? Name the disease which can be detected by this method. So what is the principle? The principle is based on antigen-antibody binding. So in ELISA, usually what happens is that, suppose there is HIV virus. I know that in human being's body, in response to HIV virus, one particular type of antibodies are produced. So we synthetically made that antibody and we are keeping it here. These are the antibodies. Okay, now I am taking a blood sample. In the blood sample, there will be antigens. If at all that person has HIV, there will be antigens, right? So when I am putting the blood sample, the antigens would be coming and binding over here. But still I would not be able to see it. So for seeing it, what do I need? I will take another antibodies, which is also something that I chemically made, and I am tagging this. I am going to tag this antibody with some coloring substance, which will change its color. So this antibody will, if it comes and binds over here, my dear children, and if I add the substrate over here, it will change the color and show that, hey, this person is having the antigens for HIV. That means this person is HIV positive. Okay, so this can be used for detecting HIV, my dear children. This is the method. So how do you write it? Enzyme-linked immunosorbent assay, that is ELISA, works on the principle of antigen-antibody interaction. A specific antigen or antibody binds to its corresponding counterpart. So you can either use antigen or you can either use antibody. There are different type of ELISA. I just told you the one where first you are using the antibody. Okay, this interaction is detected using an enzyme-linked antibody. Enzyme-linked antibody is there. Okay, I told you it is linked with something, a coloring agent. Basically, this is an enzyme, and then when you add the coloring agent, this enzyme would be changing the color of the whole thing, substrate, which produces a color change, indicating the presence and the amount of the target molecule. So the color is way too much, that means the amount is way too much. That is how you can detect it. And what is the disease? The disease is HIV/AIDS. Next question. Why are meristems preferred in tissue culture? Why are meristems? Meristems are the ones that has continuous growth. They keep on dividing. So why do you think they are preferred in tissue culture, my dear children? In tissue culture, what do you do is you take some part of the plant and you make a new plant out of it, right? So why do you take meristem? Because meristem, my dear children, especially when you talk about apical meristem, that is at the apex of the plant, and also axillary meristem, which is that towards the axillary position, both of them, they are free of viruses. They are free of viruses, which is why they can be used for the process of tissue culture. So your plant would not be affected. So meristem is free from viruses. Meristobiotic cells have capacity for cell division, which is why those are being used in tissue culture, especially apical and axillary. Remember that. Going ahead. How did an American company, Eli Lilly, use the knowledge of rDNA technology to produce human insulin? So you know what? In the older cases, what was happening is that people were taking insulin from pigs and cattle, but that was not really good because some people were allergic to it. So Eli Lilly thought that we have to actually take the human genes and then produce the human proteins. That will be the best way to do that. So they took the human gene for insulin. But the problem with the human gene was that the gene was producing a peptide, okay, A polypeptide, B polypeptide, and something that is known as C polypeptide. So this is the A1, this is the B1, and this is the C1, which is there, which connects A and B. So this is pro-insulin version. It is not active version. In the case of human beings, what happens? This is being cut. The C polypeptide is being cut, and then A and B is being joined to form the insulin in the body itself. But that was again a problem. If we put this gene inside a bacteria and produce it, there won't be things in the bacteria to cut the C protein and stuff like that. So they thought that why not? Let's do this thing. Let's put the plasmid with A gene over here, the gene that codes for A polypeptide over here. Then let's take another Escherichia coli, and then let's take another plasmid and put B polypeptide gene over here, so that it produces A and this will be producing B. Okay, now the product was taken, A and B was taken, and in laboratory condition they were joined with the help of disulfide bonds. So this is how the human insulin, that is Humulin, was made by Eli Lilly. Okay, so how do you write it? So the gene for human insulin was isolated from human cells. Insulin is made up of two polypeptides, that is A chain and B chain. I'm talking about the mature insulin, the working one. Okay, insertion into vectors. Using recombinant DNA technology, the genes for A chain and B chain of insulin were separately inserted into plasmid vectors. They were put inside the vectors. Now, transformation into bacteria. The recombinant plasmid carrying insulin genes were put back into the Escherichia coli. Right? Now, production of insulin chains. The bacteria were cultured in large bioreactors and they were producing A and B chains of humans separately under controlled environment. Purification assembly. The two polypeptide chains were then taken, purified, and they were combined to form the human insulin. And that's it. That's all. And now you have one of the applications of biotechnology is to get pest resistant plants. Justify the statement with the reference to Bt cotton. So Bt cotton, as in it is having a gene, that is Bt gene, Bacillus thuringiensis gene, that is cry gene, my dear children. I told you the cry gene in Bacillus thuringiensis is responsible for producing toxin, but this toxin is not active. This is not active. It had to be made active. But just before that, I think, okay, how many questions are there? Okay, there are just 2 questions, and then I need to give the studio to Aishwarya ma'am, which is why I'm just speeding this up. Okay, so there is cry gene and there is toxin being produced over here, right? This gene, this toxin was not inactive in the bacteria's body, but it will be active in the insect's body because it needs an alkaline pH and it is found in the insect's body. So why not put this cry gene into the plant? Now when the insect will come and eat the plant, obviously the insect will be dying because still it will be producing the toxin. The plant will also be producing the toxin. So that was the use of Bt cotton, and the plants would be stop eating it. Sorry, the insects would be stop eating the plants. So how do you write it? Some strains of Bacillus thuringiensis produces protein crystals, that is insecticidal proteins that kills insects such as lepidopterans, coleopterans, dipterans. Protein crystals form during certain phase of their growth. Okay, the cry gene cry1AC and cry2AB from Bacillus thuringiensis coding for toxins are transferred to cotton plants. The proteins encoded by these genes control the cotton bollworms. Bt plants express the Bt toxin gene by producing toxin that provides insect resistance. Bt protein exists as an inactive prototoxin. Insect ingests the inactive toxin, converts to active at an alkaline pH. That is how they work. And now the toxin will be binding on the surface of the midgut epithelial cells. Important question for MCQ. Okay, epithelial cells of the midgut. Okay, and cause cell swelling and lysis and then later die. So you can write it in your own way, make it easy for you to understand and write it in the own points. You just have to maintain prototoxin. You just have to maintain the name of the genes. Okay, and what do they kill? Cotton bollworms, corn borers, things like that. Okay. Next, how is ADA deficiency cured by gene therapy? How is ADA deficiency cured by gene therapy, my dear children? ADA deficiency as in adenosine deaminase is not there in their body, then it is not being produced because of deletion of that particular gene that codes for this particular enzyme. Now, how can that be cured is that basically, okay, by gene therapy it can be cured by basically taking my lymphocytes. I have lymphocytes in my body. I'll be taking that lymphocytes out and basically with the help of retroviral vector. In the retroviral vector, I will be inserting the gene that is coding for ADA. Okay, a complementary DNA is what I call it. A complementary DNA will be inserted in a retroviral vector. Now this retroviral vector will be affecting the lymphocytes, and when it affects the lymphocyte, it will be transferring the cDNA. Now what do? Basically, I take this lymphocyte back again and put it inside the human body. And tada, we have lymphocytes which has the gene for ADA. But again, you have to do this repeatedly. You need repeated infusions for that. Okay, so how do you frame this answer? Let's see. So the first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. This enzyme is crucial for immune system to function. The disorder is caused due to deletion of the gene for adenosine deaminase. In children with ADA deficiency, it can be cured by bone marrow transplantation, or in others, it can be done, enzyme replacement therapy can be done. But the problem is that they are not completely curable. Both these things are not completely curable. So what we can do, we can use gene therapy. Over here, the first is, at first step, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA, that is complementary DNA, using a retroviral vector is introduced into these lymphocytes, and then However, as these cells are not immortal, and then basically these lymphocytes are put back into the patient. Now, since these cells are not immortal, they will be dying, right? So we need periodic infusions. Now, one very important thing is that what you can do is the genes which are there in the marrow cells, okay, which in the marrow cells you would be having this genes coding for the ADA, okay. So what do? You take these genes and put it into embryological cells, the embryonic cells. If you put into embryonic cells, obviously the embryonic cells has a potential to divide, and basically all of the cells would be having. But for that, you should be knowing the condition beforehand itself. Then only if you have an access to this, if you know that this person has this disorder, then only you can treat it. So that would be a permanent cure. Apart from that, all of the thing is not a permanent cure. It's just there to basically let that organism or human being survive. Okay, so those are the things for ADA. Now we come on to the chapter that is ecosystem, which carries 4 marks, and there will be only one 3-marker question from this particular chapter. Okay, but before going ahead, let me just check with Aishwarya ma'am if I can go ahead with the recording or should I need to stop. Okay, I'll tell you. Okay. So basically, let's continue. So over here, ecosystem is there, my dear children, and you know what? We are really working so hard. We guys are going like back to back recording for providing you the content ASAP, because we know that PU2 exams are over here. So I hope every one of you who is watching this video is taking this seriously and studying with us properly. I'm not talking about just Jasmine ma'am, but there are other teachers also who are working really hard for you. Just please acknowledge that, tell them that they're doing a good job. That's a good thing to do, right? And also you guys study. Chalo. Ecosystem. Let's see 3-marker questions. First question. Represent diagrammatically the pyramid of number in a terrestrial ecosystem. Very simple question. In terrestrial ecosystem, the number of producers would be more, plants would be more in number. Okay, and the number of primary consumers would be comparatively lesser than plants. Okay, so I have a picture over here from your NCERT. So the producers which are there, obviously trees and all will be more. Then you have consumers such as rabbit and all. So rabbit will be comparatively lesser than that. And then if you're talking about secondary consumers, okay, such as who eats rabbit? Let's take a snake as of now. Snake is rabbit. So snake number will be less. If you talk about eagle, their number will be more less. The ones who eat, what say, snake, their number will be way too less. So if you go, it will be an upright thing that you get. This is a pyramid. Pyramid of number, you have to mention the number of organisms, my dear children, and it has to be in upright. It should not be inverted. Got it? Okay. Oh, this is what I have explained, but you do not have to explain. You have to just diagrammatically show. You will get this PDF, don't worry. Now over here, represent. Okay, so I have basically put questions over here. I'll just give a gist of all this thing. So these are actually 5-marker questions. Okay, so that is the only 3-marker question that I got from the previous years that could come, which is why I thought, now suppose the 5-marker question is broken down and given in 3-mark format, then what will you do? So which is why I put those questions also. So represent diagrammatically, that has been done. This is a 3-mark question. Now here, an example for grazing food chain is given below. Grass, then grasshopper, then birds, and then man. Graphically represent this food chain through pyramid of energy and write different trophic levels with energy levels. So my dear children, pyramid of energy, that means it will be always upright, and the first trophic level, you have to write what? A trophic level and also energy content. First trophic level is by grass, and the energy content, suppose it is, how do I write it? Where do I have space? So in exam, write it properly. Okay. I have a new thing that I studied very recently, that is this thing. Apparently, but this does not have. Oh, that's a very cool thing. Okay, just a second. Okay, we'll do it here itself, but we'll erase all this thing and we'll write it properly. So you'll see over here we have trophic level 1, which is by grass. Suppose it has 10,000 kilojoules of energy. Now going ahead with the next trophic level, which is grasshopper. Okay, so this is T2, and how much energy will be transferred? Only 10%. So that means 10% of 10,000 is what? 1,000, my dear children. Okay. Now the grasshopper will be eaten by birds. So this is T3, that is birds, and it will be 100, my dear children, because again 10% of 10,000, so of 1,000 is what? 100. you have to write kilojoules, 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 okay? And then you would be having man over here, that is occupying T4 level, my dear children, and this would be 10 kilojoules, only 10% of 100. So these are the things that you need to remember. So this is the producer, this is what the primary consumer which is there, this is the secondary consumer, and this is the tertiary consumer that you have. Cool. Now, pyramid of energy is always upright, can never be inverted. Give reasons. Because, my dear children, it would lead to ecosystem instability. How can you, organisms occupying higher trophic level if they are feeding on lower trophic level? That is not possible at all. Okay, it goes up. This is obviously going to go up. So, pyramid of energy is always going to be upright. It can never be inverted. In that case, the ecosystem, whole ecosystem would not be stable at all. Okay. So, the one that has, you already have less energy over here. So, what if you take that energy also from this guy, what energy will be there in this particular trophic level, you get it? So, the one who has more energy will be obviously giving those energy to the next organism rather than this one. Okay. Now, what do you have? So, mention the, I will be explaining the same question when I deal with 5 marker questions. Okay, so don't worry about this, just giving it, just because if at all you are someone who is just going reading this and going, then I don't want to disturb you. So, question number 16, mention the functional components of ecosystem. Mention the functional components of ecosystem. What are the functional components? Basically, you have productivity. Okay, all these things, small things which are there, productivity and everything you have to mention over here. Now, what is trophic level? Trophic level is the level occupied by an organism in one ecosystem. That is trophic level. And what? Ecological pyramids have limitation. Justify the statement with 3 reasons. Ecological pyramids have, these pyramids have limitations. Why? Because it is just considering food chain, not food web. Okay, so there can be multiple organisms, and you are not considering decomposers over here. Decomposers also play a major role, but that is not mentioned in the pyramids. So these are the limitations that you can write. But apart from that, as I said, I will be explaining in a much better manner when I talk about the 5 marker question. Okay, chalo. So these are the questions you have to take into consideration. Okay, suppose a 3 marker question comes out of it, so I don't want to go astray. Now, biodiversity and conservation, my dear children, you have basically 4, so the whole marks is 4, but you can get a 2 marker question coming from it, 2 marker question, and also you can get 2 MCQs coming from this particular chapter. So, briefly explain the significance of David Tillman's long-term ecosystem experiment using outdoor plots. So, he thought that basically when he was doing the experiment, he found out that when he grew certain more plants, okay, the ecosystem was more stable. That was the thing. Just a second, just give me a second. Yes. So yes. So we were talking about the experiment. I will be directly showing you the answer for better understanding. So see over here. So David Tillman's long-term experiment said that he conducted using outdoor plots, and he demonstrated this thing, that is, increased biodiversity enhances the productivity. Ecosystem with higher species diversity show greater primary productivity. They show primary productivity, and plants were able to produce more things, and obviously the organisms depending on it will be obviously able to survive as well. So as different species utilizes resources more efficiently and complement each other, obviously the increased biodiversity would be increasing productivity as well. So if you have more plants and if you have more animals, all the animals have different resources to eat, my dear children. Obviously they won't be competing for each other, and this will be obviously increasing the productivity as well. Now there will be stability also that would be increased, that is, diverse ecosystem are more stable and resistant to disturbance like droughts or pests. For example, there are certain plants that I have which are they have tolerance to salinity. Okay, they can survive in very salty condition as well. But suppose there were just plants who were not able to survive through, there were just similar kind of plants all over, we do not have diversity. So don't you think all the plants would be dying, since because they do not have the capacity to be tolerant. Now because we have diversity over there, they are able to survive, and probably they can reproduce and increase their number. So obviously stability is also being maintained. Now resource utilization, biodiverse plots utilize nutrients and sunlight more effectively, minimizing the wastage and ensuring better ecosystem. So there is no wastage because you have more diversity. So everything that is there, every resources which is there, it is being utilized very well. Okay. So yes. Now, name any 4 recent extinct organism as per IUCN Red List. So I have pictures as well, I think, but let's see. So dodo, from Mauritius, quack from Africa, thylacine from Australia, stellar sea cow from Russia, and 3 subspecies of tiger, that is Bali, Java, and Caspian. So these are the 4 things. Any 4 things you can write as an extinct organism as per IUCN Red List. I think I had picture. Yeah, so this is dodo, that is from Mauritius, my dear children, and this is the thylacine, or this is a stellar sea cow that I have. Okay, yes, I think that is the thylacine which was there from Australia. Okay, but I have to be sure about the picture once more. So dodo is from Mauritius, quack is from Africa, thylacine from Australia, and stellar sea cow from Russia. So you would not never forget stellar sea cow. So it is in the sea, it looks like a cow, which is why it is known as stellar sea cow, and it was overhunted, which is why it got extinct. Okay, and dodo is a cuto over here, very cute, right? This one. Okay, and yes, this is thylacine only. Yes. Now, explain biodiversity, define biodiversity, and write any 2 type of biodiversity. So let's see the answer. Biodiversity refers to the variety of life forms which are there on earth, my dear children, which include different plants, animals, microorganisms, and everything. So it includes variation within species, between species of the ecosystem. You have different type of diversity, genetic diversity, and also species diversity. Genetic diversity as in it refers to the variation of genes within a species. Example, different varieties of wheat or rice. Okay, within a species you have different wheat type, you have different rice type, you get it? So it shows what? Genetic diversity. Now, species diversity is what? It refers to the variety of species within a particular region. For example, in one particular region, I have different species of birds, insects, and things like that. So, diversity of species in a rainforest including birds, insects, and plants, everything together. So, this is species diversity. Okay. Now, what do you mean by endemism? Explain any 2 causes for biodiversity losses. So, endemism means species which are just found in specific regions. You cannot find in any other region. So, these are known as endemic species. They are very unique to that particular area. Okay. So, endemism basically is a species confined to specific region and not found anywhere else. And 2 causes of biodiversity loss, basically habitat loss or fragmentation. You are destroying the area where the organisms are living. Okay, so destruction of natural habitats due to deforestation, urbanization, you are making big buildings over there. Agriculture or industrialization reduces the space for species, leading to what? Extinction. That is there, such as clearing forest for farming has led to loss of many species. Now, overexploitation, yes, excessive use of resources like hunting, that is how the stellar sea cow lost, right? We do not have it now. It was there in Russia, but we do not have it now because of the overexploitation. We kept on hunting. Now, fishing and logging depletes populations faster than they can recover. Example, disappearance of stellar sea cow. Okay, and then alien species invasion. For example, because of the beauty and things like that, we basically we take things from other countries and we bring it over here, such as Ecornia. Okay, Ecornia, the water hyacinth. So what happened was it's just spread in the water, my dear children. Now when it spread, basically it is covering the upper layer. Do you think the light can get inside? No, the light was not getting inside. So again, that was a big problem that was being caused. The plants which were there below, they were not getting enough light. And apart from that, when this particular plants, they used to die, they used to go sediment. It increases the organic matter in the water, creating algal blooms and a lot of other things as well. So that is alien species invasion that causes a lot of trouble. Now co-extinction, as in one organism is getting extincted, so other organism which is totally associated with it, that will also get extinct because of the same reason. Okay, so that is one thing. So these are the causes of biodiversity losses. There are 4. I just explained 2 because you are asked about 2. Now, give any 2 examples of sacred groves in India. What do you mean by sacred groves? So, I am really happy because we are completing it finally. And sacred groves, my dear children, are areas which I have some religious connection to those areas. And not just me, a lot of people have it. So what is happening? I am safeguarding that area, but also I am safeguarding the organism, the plants which are in there. So aren't the animals and the plant species also getting safe? Yes. So these are known as sacred groves, my dear children. The very famous question comes, what is there in Madhya Pradesh? It is Chanda and Bastar. Bastar of Madhya Pradesh, they are acting as sacred groves. You have Khasi and Jaintia hills in Meghalaya, very, very important. Aravali hills of Rajasthan, Western Ghats of Karnataka, Sarguja, Chanda, Bastar areas of Madhya Pradesh. My dear children, you can expect an MCQ from this thing. It's a very, very, very important question that you see over here from this particular chapter. Okay, Khasi, Okay, Khasi and Jaintia Hills in Meghalaya, Aravalli Hills in Rajasthan, Western Ghats of Karnataka, Sarguja, Chanda and Bastar of Madhya Pradesh. You have to remember it anyhow. Okay, cool. Now, there are many reasons for conserving biodiversity. Briefly explain the reason for conserving biodiversity from a narrowly utilitarian point of view. What do you mean by narrowly? So, there are many reasons, and one argument was narrowly utilitarian point of view. That means basically we should be conserving the nature because it provides us with good products such as fruits, such as drugs, the plants. I'm talking not about the drugs as in weeds, okay? So drugs, that is basically medicinal products that we get. So we should be taking care of the nature because it is providing us with certain products. Now there is another point of view also where we would be protecting nature because of the services that it provides. So this is not that. This is narrowly utilitarian point of view, my dear children, where you're just talking about basically the products that we get, such as food, such as medicine, such as industrial products, economic value and all those things. Food example is plant, animal and microorganisms that are sources of food. Medicine, many medicines are derived from the plants and microbes. Industrial products, you get raw materials for industries including timber, fiber and all, everything. Economic value, many species support livelihoods, trade and tourism. So again, this is the products that we get. So from narrowly utilitarian point of view, biodiversity is conserved because it directly benefits human by providing essential resources and services, my dear children, and this is how you get 3 marks from the question. Now, what is ex-situ conservation? Mention 2 example. Ex-situ conservation as in you are not conserving organism in their habitat. You are making them move out of their habitat and then you are conserving it. This is ex-situ conservation. Okay. Now, example is, for example, zoological parks, zoo. You're taking the animals, putting in the zoo and conserving it over there. Botanical gardens, you're taking the plants and putting it over here. These are the examples of ex-situ conservation. So zoological park, botanical gardens, wildlife safari parks, your conservation of gamete by cryopreservation, seed bank, all of it. It's basically wildlife safari park is different from wildlife sanctuary, okay, please remember it. Now, ex-situ conservation, threatened plants and animals are taken out from their natural habitat and placed in a specialized setting where they can be protected and given special care. Okay, so that is ex-situ conservation. Now, mention 4 evil quadrate which can cause depletion of biodiversity. Among vertebrates, which group of animals has highest number in global biodiversity? That is also a question. So, first thing, evil quadrate, which is there, my dear children, over here. Evil quadrate refers to the 4 major causes of biodiversity depletion. We already studied this, that is habitat loss and fragmentation is one, overexploitation is the next one, where stellar sea cow is an example. Then alien species invasion is where non-native species are being put over here, such as I told you, water econia, that is water hyacinth econia, okay, that is there. And the co-extinction, when one species become extinct, the depending organism also become extinct. Now, extinction of host plant can lead to extinction of dependent pollinators. So there's a plant and there's a pollinator which is dependent. You know yucca and moth, we know about it, no? And we studied in the chapter that is sexual reproduction in flowering plants itself. So if there is no yucca, there won't be that moth also which will be there because they are cohabiting, right? So that is the thing. Now, among vertebrates, as in the ones that have vertebral column, my dear children, okay, so which are the group of animals which are higher number? So you have fishes, you have pisces, you have amphibians, reptiles, aves and mammals, right? In them, the highest is been occupied by the fishes. In vertebrate category, the highest is fishes. If you're talking about invertebrates, it is insects. Okay? Now, mention the 4 evil quadrate. I already spoke about that, and this is the fishes. So, fishes, then you have reptiles in more amount, then you have birds, or basically birds, and then reptiles, then amphibians, and then mammals which are there. Okay. Alien species invasion caused decline or extinction of indigenous species. Justify the statement by giving 3 examples. So, first was water econia, but let's see the other examples also, okay. So, Nile perch introduced into Lake Victoria in Africa led to what? Extinction of ecologically unique assemblage of more than 200 species of native African cichlid fishes in the lake. So, Nile perch, I will show you the Nile perch which is right over here, my dear children. This Nile perch, when it was introduced, my dear children, what happened? The native African cichlid fishes which were there in the lake, almost 200 species of them, they got extinct because of the things. Okay, so it's because of they were introduced. So, this is invasive. Right, they were introduced, they were alien species which were introduced and they caused extinction of the indigenous as in our own species. Now carrot grass, that is Parthenium, lantana and water hyacinth, all of them are examples of plant invasive species, my dear children. Okay, so over here you have the pictures. This is lantana, this is Eichhornia, and this is the other one, Parthenium. Okay, I think, okay, just give me a second, I'll just confirm which picture is which one. Okay, yeah, basically I was correct only. So this thing that you see is Parthenium, my dear children. You might be seeing in the wild area, in the garden area and all, you will be finding this thing really going fast. So this is very toxic, okay. And this is Parthenium, and this is lantana, okay, and this is Eichhornia, which spreads very rapidly, about a hyacinth. So yes, now you have African catfish, that is Clarius garipinus, my dear children. So they were pretty good, they good looking, so they were taken for aquaculture purposes, but they posed threat for the actual indigenous catfishes which are there in the rivers. And now this is again an MCQ question. So I think I have Clarius garipinus picture. No, I do not have it, so I cannot show it. So these are the things that you can remember. I showed the picture as well because with the help of pictorial representation you remember things better, right? So yes, so Nile perch is there, okay, it was a fish, it was introduced and into Lake Victoria, my dear children, in East Africa. But what happened, the cyclid fishes which were there, 200 species of them, they lost their life because of this extinction happened. Now carrot grass, that is Parthenium, lantana, which has beautiful flowers, and water's hyacinth, all of them, my dear children, they are invasive species of plants. Now African catfish, that is Clarius garipinus, for aquaculture, obviously it poses a very serious threat for indigenous catfishes which are there in the rivers. Now with that we complete the chapter biotechnology principle and processes. You might have seen that I missed it because there are no 2 and 3 marker question from that particular chapter that is going to come. And similarly organism population, again a 5 marker or 6 marker chapter, my dear children. It won't be giving you any 2 marker or 3 marker question. There will be 5 marker or 1 marker question. That's all. And with that, we are completing all the chapters of our biology, and I hope this video was very sufficient for you to study. I hope you just give your 100% into this and do not panic seeing the number of questions and everything. You have come this far, you have revised, and that's more than enough. Okay, so just keep this information in your head and everything is going to be sorted. Yes, and do not forget to like, share and subscribe, and do let us know how much you love us in the comment section. So that's all. Bye-bye. Take care.